Hello, hello. Hello, hello. Sound check one, two. Hello.
हमने एक बार ट्राई कर
Hello, hello. Sound check one, two. Follow, follow, follow.
Good morning, everyone. I, Shweta Pandey, welcome you all on behalf of the organizing committee for the day 2 of the conference on antimicrobial resistance and human microbiome, jointly organized by the DPT Translational Health Science and Technology Institute and the NIH, National Institute of Allergy and Infectious Diseases, under the aegis of Indo-US Vaccine Action Program. May I request all of you to please keep your cellular devices on silent mode, and for those joining online to please put yourself on mute as we will be starting the program now. Starting with the first scientific session of today, we have talks on the theme homeostasis and dysbiosis of microbiome in health and disease. I humbly call upon the session chairs Dr. Shinjini Bhatnagar, Dr. Avinash Bajaj, and Dr. Ganesan Karthikeyan to the stage. Dr. Shinjini Bhatnagar is a distinguished professor who heads maternal and child health research at THSTI. Thank you, ma'am, for joining us today. Dr. Avinash Bajaj is a professor at the Regional Center for Biotechnology, Faridabad, India. His research focuses on developing next-generation nanomedicines for cancer immunochemotherapy, understanding the role of sphingolipid and ganglioside metabolism on therapeutic interventions, and developing next-generation biomaterials for anti-inflammatory, antimicrobial, and gene therapy applications. He has more than 70 publications in high-impact journals, which also include his work on cancer immunotherapy. It is our pleasure to have you here today, sir. Dr. Ganesan Karthikeyan is a clinical interventional cardiologist and a professor of cardiology at the All India Institute of Medical Sciences, New Delhi, India, and a senior international fellow of the Population Health Research Institute at McMaster University, Canada. He is an associate editor of Heart, deputy editor of Open Heart, and co-editor of the European Heart Journal. His research is mainly focused on cardiovascular diseases affecting low and middle-income countries, including valvular heart disease, particularly rheumatic heart disease, mechanical valve thrombosis, anticoagulation, and indigenous drug-eluting stents. It is a pleasure to have you here with us today, sir. In this session, each talk will be of 15 minutes. After the talk of all the speakers, there will be a 10-minute long question and answer session. I request the audience to kindly reserve their questions till the end of the session. I now request the session chairs to introduce the speakers and call them over for the talks. One of our speakers, Dr. Saurabh Datta, will be joining us online.
Good morning, everyone. We're going to start the next day of this very interesting workshop, conference. I'll hand over to Avinash to introduce the first speaker.
Good morning, everyone. Good morning, everyone. So today we have first speaker, Dr. ARHM. He's a scientist at Stanford University, and he has done his PhD in computer science, where he has focused on epigenetics and chromatin folding at Weizmann Institute. And he has done his master's degree, very interestingly, in computer science, where he was focused on developing the computational approaches to study the three-dimensional properties of the protein channels. And currently at Stanford University, he's working on experimental and the computational approaches to study the evolution of natural microbial communities under selective pressure, such as antibiotics. Yeah. Welcome, sir.
And good morning, everyone. First, I'd like to thank you for that nice introduction, and it's great to be here. And the first day was fascinating, and I'm looking forward also to all the talks that will be here today. So I am going to tell you today a simple story compared to many people were reviewing lots of work. This is a very straightforward story of evolution inside in vivo in people. And when we think about AMR, or antibiotic resistance, we know that usage drives resistance, but we know it from a very, very broad scale. So if in a certain area or region, we're prescribing some type of antibiotics, and then over months, we will see resistance arising. So that's a very coarse or gross association. And so that's, for example, shown here in this study where they compared seasonal changes in resistance and correlated it with usage. What I wanted to do was try to track resistance as it's happening in real time inside people. So that's actually not that easy to do. So evolution is rarely documented in clinical settings. What usually happens is bacteria are evolving as they're moving around the world, exposed to understood conditions and exposed to all kinds of antibiotics. Eventually, they end up in a specific patient, in a specific situation, and there you see their resistance. So the entire process of the evolution of resistance is actually left understood and uncharacterized. So my broad aim was to quantify the evolution of resistance in people during the course of antibiotics. So that sounds something pretty straightforward to do. So we know already a lot about the evolution of resistance. And we know that from in vitro studies. So it's very easy to take your favorite bacteria and to expose it to antibiotics. Over time, you start from a clonal population, and then as you are exposed, as the selective pressure is working on this population, you will have a de novo mutation, and it will sweep in the population, resulting in something called a selective sweep. So those are very easy to characterize in vivo. The question is, can we do it in an uncharacterized mixed population, such as the gut microbiome of people? Which is a much more complicated problem. And the reason it's a complicated problem is because a community of bacteria is not a single population. It's actually a mixture of many populations living together. And there's horizontal gene transfers moving, shuttling genetic information between the genomes. So it's much harder. This picture here depicts the idea that the genomes have an overlapping in their genetic components. The genes overlap between the different populations. So you have to put that apart. You have to resolve genomes. It's much more complicated to do it from actual undefined communities. So there's going to be a whole workshop dedicated to that, and I'm not going to dive deeply into that. But let's go forward into our experimental design. So what we actually did was a pretty simple experimental design. We took 60 healthy volunteers. We exposed them to ciprofloxacin for 5 days. Ciprofloxacin is a type of fluoroquinolone, and a lot is already known about the resistance mechanism of fluoroquinolones. And many people, actually, several talks discussed that yesterday. And for each subject, we took 16 samples depicted there in the bottom. And so it's from 60 days before to up to 78 days after the antibiotic exposure. And we sequenced them deeply with 300 million reads per sample. And reconstructed using pretty standard methods that, as I said, you will learn in the workshop, genomes. And these genomes, I call them consensus genomes. Some people call them MAGs. And each genome, it's important to remember, represents a population of a specific species inside a specific subject. We have over 5,000 of those. And each one of them, I say it represents a population because you can have multiple colonization events, and even a single colonization event starts clonal, but then you have a population structure, which is exactly the interesting part that we want to study. Okay, so from this experimental design, the first thing that we wanted to do is ask very, very broad questions on the population level. And when we look at the population level, we take our 5,000 genomes, so it's about 90 genomes per subject. And we can ask how they change in their abundance over time. So that's a very broad question. And here we see clusters. I clustered them using k-means into 100 trends. Time is in the x-axis, the clusters are in the y-axis. You can see the color represents the relative abundance over time. So there's a very beautiful cluster that appear. You can see on top, for example, extinction events in which bacteria went under detection level and never came back. Then you can see more drop events in which populations dived, but then drove back up. You can see blooming event, in which they were transient during antibiotics. So you have all these processes occurring. So that just shows to us that there's a strong selective pressure on our systems, and bacteria respond in a differential manner. Bacteria here in the middle seem to be either tolerant or resistant or relatively resistant, because all of these assays are tolerant, are relative in nature. So we don't have an actual measurement. And that's what's kind of interesting about this type of assays. We don't measure mix or susceptibility for any of these commensal bacteria, and we know very little about them to begin with. But to summarize, if you compare each dot in this plot is one population, you compare pre and post abundance levels, then two things come out. First of all, extinctions are pretty rare. Only 10% of bacteria disappeared during our disturbance. So we have some kind of robustness. And more than that, the ones that did survive, they keep their abundance rank. So they actually jump back to the level of abundance that they were before. And all of these show the test to the resilience of the human gut. So on the surface of populations, everything seems to be pretty stable. We have this people that are exposed to antibiotics, asserts selective pressure, and then the population just bounce back up. It looks like nothing happened. And here exactly is where I wanted to go in and ask, if you go dive deeper to the nucleotide level, can you actually see what this selective pressure actually creates in these populations? So that's basically our setup. Okay, so going to the next stage, you actually want to go beyond this very broad populations, and we want to go to the level of variance. So the way we compute variance is by mapping our reads from each of the 16 samples back to our genomes. And after we map them, then we can see a comparison between, if, for example, in this imaginary point, you compare pre and post, you can see that this one position in one imaginary gene has a substitution from A to G, with a little a staying behind. And then you can ask yourself, okay, well, this is probably associated in some way with resistance or some kind of selective pressure. So immediately you think this has to be some kind of, has to have some kind of fitness significance. And I'll show you in the next slide why that's actually not the case. Most of these are what are called spurious variants, which have nothing to do with resistance. That's really what's been hampering, I believe, the study of metagenomics and in the context of these type of selective pressure studies. So the broadest way to look at this problem is to think about the idea that you have many populations that are mixed together, and they can have overlapping genomes. And what you want to do is you want to separate what are true polymorphic sites from spurious sites. And I'll demonstrate it with a simple example. Here you have two populations. On the left, you have just one species, and that species has just two strains separated by 3 mutations. And so these are actually competing. These 3 mutations, as you can see over time, black's taking over, which means that these 3 mutations might have some kind of fitness, association to fitness inside this population. That's great. That's the true polymorphic site that we're looking at. But there's another case. This other case, you have 2 different species altogether, the blue and the green. What they have is some kind of a shared element. Think, for example, some kind of a mobile element, like a transposon. And that transposon, or that mobile element, can have also a polymorphic site. So if you look just locally at the site, it looks exactly like the same. You have this switch between white and black. And so you might attribute that. But here, on the left, it's completely spurious. Why is it spurious? Because these 2 variants are not competing on the same background. They're actually reflecting a competition between different species. So this is not really that interesting for us when we want to go to understand fitness and the genotype-phenotype associations. So, and without going into the technical details, we developed a set of statistical methods that manage to separate true polymorphic sites from noise. And the main trick is just to look at not only the abundances of the specific site, but also an abundance of the entire population. So here, for example, the true alleles, their sum needs to sum up, their sum of abundances needs to equal the abundance of the population that they belong to. While if it's a spurious variant between two populations, then each of the alleles will have the same abundance as its own population. So this is all wrapped up inside the tool that you can find on GitHub called Polypanner. Cool. Okay, so first thing we want to do is test this, and we tested it on simulated data. So simple lessons from what we saw from simulated data, which we just know everything. We have a ground truth, so it's very easy to understand what's going on. First of all, the problem is extremely pervasive. We can see that due to assembly issues, which is basically what's underlying all of these issues, all of these spurious problems, you have an order of 100 spurious sites per megabase of genome. And this is regardless of the coverage, regardless of the complexity. Even if you have clonal populations, you will still have that level of a problem. And our approach reduces the density of spurious variants by 100-fold. So we're basically cleaning that type of noise pretty well. And there's a price to pay for this type of sensitivity. And the price is that you need to sequence more deeply. And that's something that's becoming less and less of a problem as sequencing costs drop. But you need to sequence, for example, to detect SNPs, you need to sequence over 100x for each of these microbial genomes, 100x summed across all of your samples, as opposed to, so you need that level of sequencing, which is an order of magnitude higher in order to actually detect things. So we have this method that's precise, and also has some sensitivity issues, which is inherent to this approach. Cool. So what we did is we go back to our data. I want to go back to our beta, and we applied it to our cohort of over 5,000 genomes. And we found over 2 million polymorphic sites. And those can be clustered according to linkage into an order of 10,000 strains, similar to the strains that I showed you before, because they're not all different strains. They're not completely, they're linked together. And what we found is we found over 500 populations in which there was one or more sweeping variant. And a sweeping variant is a variant that switched from under 20% to over 20% to over 80% during the antibiotic exposure. And I just want to show you one example out of these 500, and this is a nice example here. So it's a Meganomanus funiformis in a specific subject, and you can see it has 4 strains, and you can see their abundance over time. And you can see during antibiotics, first there's a crash, so it's really the abundance just disappears, and then it blows up. And after the population recovers, there are the 2 different strains that appeared, which have the plus here. And these strains are separated by exactly 3 variants. So these strains are very, very similar to each other. Very few variants play part here at all. And so if you just look at this, you say, well, these 3 variants probably have something to do with resistance. We can't tell which one is it. So we have 500 examples of this. And here, the next step in order to understand where actually selective pressure is working is to think about hitchhiker and driver. So each selective sweep is typically driven by only probably one mutation that has a strong fitness advantage, and the others are actually hitchhikers, and they're neutral or even slightly deleterious. So in order to actually find where the selective pressure is, we turned to converging evolution. And the way we did it is, at this point, we started summing up data from all of our subjects into genes, and through genes, we moved to KEGG orthology, which is a functional notation of genes, and we just counted how many KOs, how many gene variants map to each KO, and we compared that to a background model that is just, genes are just, the variants are just randomized. So the first thing that popped up right on top is gyrase. That is not surprising, as you heard yesterday, and it's also very well known. Gyrase is the target of ciprofloxacin. Therefore, we expected to see it here, but it's nice to actually see it. And it's really widespread. So 41 different commensal species had some kind of a sweep in gyrase. Specifically, gyrase, the subunit of the gyrase. And you can see the DN-DS ratio, which is a hallmark of adaptive evolution, is close to 4. And beyond gyrase, we see something which is a very dispersed signal. So there's many functional KOs that get some hits, but it's a very idiosyncratic signal in which different species probably utilize different genetic mechanisms in order to develop resistance. And so that's our analysis of parallel evolution. But we dove more deeply into GRA, and then you can see where exactly in GRA these variants map to. And you can see that they all map here to a very small area in the beginning of GRA. If you look more closely, then they actually pile up very two specific amino acids, 83 and 87, which are again known to confer resistance. That's exactly the binding site of Cipro. But what's cool about it, first of all, we just inferred all of this de novo without doing any experiment. This just came out of the analysis. But more than that, this is a chance to see how evolution of resistance through mutations in GRA actually transpire in vivo. So we can start to have quantitative measures of this evolution. And what we can see, for example, there's many measures that you can count, but probably the most interesting one is how many of the bacteria actually evolve through mutations in GRA. It's 10%. So it's a pretty significant number. Just this one short course of antibiotics, 10% evolve resistance through mutations in GRA. Then you can ask yourself, well, why do these 10 evolve resistance and not others? And I thought it's going to be 20 minutes. Is this because I'm good? Okay, just let me know when I'm gotcha. No worries. Cool. So I don't have much more anyway. So I have 3 more slides. So we got this. And so the question is basically why this 10% got mutations in GRA, not the 19 others. And for that, we define evolvability, which is the probability of a population to evolve resistance through mutations during the disturbance. And we can see that evolvability is explained by 2 very general fundamental properties of the population. The population size, which is measured by the abundance, and the decline in abundance, which is a proxy for drug sensitivity. In other words, so we defined a logistic regression model that shows that we can predict the probability of a population to get a mutation in GRA based not on its genetic composition, but it's in size. Bigger populations have more tendency to develop resistance through mutations in GRA, and also its susceptibility, which makes perfect sense and also suggests that these mutations are actually occurring de novo. And so just one small thing, this is a very good model, but it also many species deviate. So there's also a taxonomic specific signal that's worth to dig further. And in the last slide, we asked the question, well, what does it even matter? So we have these transient mutations, maybe it all goes away and there's not any issue with GRA. This is just transient during the exposure. And the answer is no. So we can see if we look at the last day, which is 10 weeks out, then we can see that most of the bacteria actually, populations keep the GRA mutations. And if we look a year out by just doing a very basic inference of projecting out, then we can see that again, more than 95% actually keep their mutations. So you get this GRA mutation and you keep it. Similar trends for non-GRA mutations. But really the bottom line here is that there is a very low, seems to be a low fitness cost for these mutations in GRA. So this is really something that to think about. And yeah, so to conclude, first of all, this manuscript is available by arXiv. You're welcome to see it. Polypolymer is also available on GitHub. And just future work would be to start thinking of other antibiotics and also to think about different selective pressures. And all of this work doesn't have to do anything with horizontal gene transfer. So there are other methods to study horizontal gene transfer, which I'm working on right now, which are Hi-C, and also you can use long reads. And for the sake of time, I would not skip the summary. And yeah, I would like to just end by thanking David Relman, my mentor and PI, and all people in my group that were involved in this. And thank you very much.
Thank you, Dr. Yaffe. That was a fascinating talk. We'll take questions at the end of the session. So it's my pleasure now to invite Dr. Shalimar. Dr. Shalimar is a professor of gastroenterology at the All India Institute of Medical Sciences, and he has done a lot of work on the microbiome along with DHSTI. His main areas of focus are on NAFLD and other diseases of the liver. Welcome, Dr. Shalimar.
Thank you, sir. Good morning to everyone. So I'm a clinician. I'm a hepatologist. I don't do all these microbiome work. It's all done by Dr. Babatosh. So all the slides which I'm going to show is his work. So if you got any questions, please ask me. Don't ask me questions on the microbiome. I don't. So that's my disclosure. Okay, so I'm going to talk to you about a disease which is called as non-alcoholic fatty liver disease and some work on the gut microbiome, which we've done with Dr. Babatosh. So what is this fatty liver disease? So we all love food, and you got such a good set of breakfasts flying outside, and we like eating all those fatty foods, drinks which are cola drinks, which are sweetened. We like drinking alcohol also. So what this all does is it all gets deposited in your liver. So when you're eating more and spending less, everything starts getting accumulated. So that's your bank. So your bank is now fat. So when this fat goes on accumulating in the liver, and when it's more than 5% of your hepatocytes have fat, that is what clinically we call as fatty liver disease. That's when the patient comes to us, which may have problem or he may be asymptomatic, right? And this may progress on to cause damage. Okay. So before we talk and more further, there is a change in terminology of fatty liver disease. The term what we classically used to be known as non-alcoholic fatty liver disease. Now, if you look up into the literature, it has been changed to a term which is called as metabolic associated steatotic liver disease. Acronym for it is MASLD. So you will not hear this term NAFL more frequently now, as the experts have renamed it as MASLD, right? Essentially, it's the same group of patients, but then we define these MASLD as NAFL with addition one metabolic abnormality. So 1 on the 5 metabolic syndrome parameters is abnormal. That's what is called as MASLD now. Why do we need to hear about this? It's globally, the data suggests 1 in 3 individuals have this entity, and I'm sure we've got friends from different countries and they know about the same situation everywhere. So globally, 1 in 3 have fatty liver disease. It's not a phenomena which is restricted to the highly affluent Western countries. You go across the globe, it's there. Now let's come to India, where we are. Even studies from India suggest that almost 4 in 10 of individuals, that's the population we're looking at, has fatty liver disease. So if I look at the people sitting out here, and I'm sure if you get yourself screened, 60% of you would have fatty liver disease. You are asymptomatic, that's why you don't have problems, but then there is excess of fat. Whether it is a problem of worry or not, that we need to see, depending upon has your disease progressed or not. So the burden definitely is there in India. So within this fatty liver big cohort, which we are talking about, we've got simple steatosis. That means there is fat. It is not causing you any problems. Like we all believe, we don't have problems. There is a group of individuals who get this inflammation. So like this fat moves on and there is a lot of inflammation in the liver, and that is what we call as NASH, or the new terminology is MASH. So problems developing in the liver with steatosis and inflammation is MASH and fibrosis. I'll come back to you. That's why I'm spending a bit of time on that. This is the problem group. This so far, from our understanding, it's not a problem group. It's there. As I talked, 40% of us have fatty liver disease. But then if you develop this, there is a problem. So why are we developing fatty liver disease? The diet, we've had a good breakfast today, and we have it. Lifestyle, we don't like exercising. We're sedentary. We like sitting. So that's one more factor which adds to fatty liver disease. Now there comes a component of genetics. Like Indians, particularly, possibly, we are thin. Contrast to our Western counterparts who got high BMI, and we still have low BMI and we get fatty liver disease. That's a paradox, right? So possibly a role of genetics, and now comes the gut microbiome, what we are discussing in this meeting. So I'm going to talk to you about in the next slides on the role of gut microbiome. And then there is obesity and diabetes. We are a hub for diabetes. And we've got almost like 10% prevalence of diabetes, 20% prevalence pre-diabetes, looking at a huge population. So even a study from down south suggests that almost 70% of Indians have one abnormality in the lipid parameters. You're looking at dyslipidemia so common. These are the persons who don't know because they're not getting tested. So there are some issues which predispose us as Indians to develop more fatty liver disease. Microbiome, you've already listened over the last 1 day or so. It's associated with lots of diseases. And now we've got even translational value of microbiome. We've got some gut microbiome, like we've got fecal microbiome transplantation coming into practice for Clostridium difficile, which is an infection in the GI tract. But there's a lot of data now coming up. And most diseases have been associated. So whether this gut microbiome really causes the disease or it's a mere association, it's a change, it's an effect, it's still evolving. We're getting more and more data on that part of how the gut microbiome plays a role. So coming specifically to the fatty liver disease, which we are going to talk about. So we've got food, what we eat, it goes through our food pipe, goes into the intestine, and it gets absorbed. So when it gets absorbed, all the blood we know moves from intestine into the liver, which actually takes care or say something like, I'll say, put it in simple language, it metabolizes the food what you're eating. So intestine absorbs, liver takes care of it, and then there is a crosstalk. So you absorb things from here, goes into liver, liver produces something which goes back to the gut, and that's how there is a sort of a communication between the two. It's something like an SMS, what you see, it's something like that, there's a talk which goes. And how does this crosstalk occur? So liver produces what we know as is bile acids. Bile acids go into the intestine and then produce some influences. So what are these? So we know that there are a lot of these microbiota, gut microbiome, which are residing in your intestine. They produce a lot of these short-chain fatty acids, which we typically known as butyrates, acetaldehyde, and there is production of ethanol. You thought you were drinking alcohol only. Your gut microbiome is producing alcohol also. So even if you're not drinking, it's being produced in your body by the microbiome. Now, this leads to a lot of changes and there are then these inflammatory mediators which are produced by the liver, which go back and affect. So in a nutshell, there is a lot of crosstalk going on between the liver and the gut. So where do these microbiota play a role? So there are a lot of these, like which talked about, these small molecules like short chain fatty acids, which in turn affect the metabolism within the hepatocytes. There is ethanol, which we know ethanol causes damage to the liver cells. Then there are these LPS, peptidoglycans, which also affect the liver cells. And then there are these effect on the Kupffer cell, which cause more of changes in terms of pro-inflammatory cytokines, again damaging liver. So gut microbiome, what we understand is possibly could be, one is influencing the production of molecules which affect the liver, and also affecting these Kupffer cells, which then play a role in the fibrosis. And possibly these bile acids and the modifications of bile acids which happen because of gut microbiome is another player. So we know gut microbiome, we've got lots of these taxas. I'll not go into details, but important to say is that this is what they produce and they may cause damage to the liver. Okay, so let's move forward. When we talk about the gut microbiome is influenced by multiple factors. Age, where do you belong to, what kind of foods you have, alcohol, drugs, lifestyle, underlying diseases, diabetic. So there are a lot of factors which play a role in this gut microbiome, and it is different in different individuals. This is data from Dr. Babatosh. He's kind enough to give me these slides, and this is what he looked at, a comparison between the Indians and the Japanese. Interestingly, the data suggests there are differences in the gut microbiome, and what he showed in the study is that Indians have more Prevotella, and in contrast, the Japanese have more Bacteroides and Blottia and Coprophocus. So there are differences. Could this be one factor playing a role? I think that's something which needs to be again explored. There are also differences in the fungi. So again, looking at the Japanese have more Saccharomyces, but look at this, Candida. This is more seen in Indians. So this is a peculiarity of, could these differences in the bacteria, fungi, possibly be playing a role? So this is data from healthy individuals. Let's move to some disease states. So if you look at, this is again data provided to me by Babatosh. It's a study where he compared individuals with pre-diabetes and diabetes in the Indian and the Danish cohort, again suggesting differences between the two populations. And this is very clear that there are differences in the gut microbiome, and could they be a player? Okay, now let's come back to the liver, what we are discussing. This is a very interesting data, which suggests that when liver tissue is taken out and then you look at into the bacterial rDNA copies, then there are also differences. So the obese individuals have lower alpha diversity and higher proportion of proteobacteria. Again, suggesting, we talked about gut microbiome, that's the stool, the intestine. Now in the liver also there are changes. Fecal microbiome. So when we look at a comparison of individuals who got NAFLD based on non-obese and obese, there are significant differences in the non-obese population between those patients who got NASH, which I told you initially is a disease state when NAFLD moves with inflammation, has more damage and has fibrosis. So there are differences in the fecal microbiome. So we know from existing literature, there are changes between the microbiome. And what this interesting study added to the literature was that when in the mice model, when you treat with amphotericin B, which is an antifungal, the amount of steatosis, see, this is a controlled diet, and when this is a mice which has been fed with Chow, and then which is fed with Western diet, there's a lot of steatosis. Can you see these white rounded spaces? This is fat in the liver. When you treat the mice with amphotericin B and follow the same steps, they don't get fatty liver. So is it suggesting possibly use of antifungals may be associated with a reduction in steatosis? Okay, so where does this virome? Again, there are data to suggest that there is significant difference in the diversity, and those fatty liver disease who got advanced disease have less of virome diversity. Okay, now coming back to the study which I want to highlight over here, or which we've done is, so this study is done by Dr. Babatosh along with his team, and especially by Ayushi. So I'm not sure if she's there, but then this is her work. So I'm presenting her work, it's not my work. So we raised questions over here is, can multi-omics profiling of gut microbiome of NAFLD provide some kind of bacterial signature which helps in differentiating? And can this provide us with some kind of mechanistic picture regarding the steatosis and the disease progression in Indian population? So that were the questions which we raised for this study. So we took patients from the clinic, is obese patients who didn't have NASH, those who had NASH, and healthy individuals, and compared their gut microbiome. So what this shows is that those patients who are obese and have NASH have less OTUs compared to the healthy individuals. So the richness over here in those who got fatty liver disease is less of the gut microbiome. And when we look at NASH compared to the obese and the healthy, the richness over here is even more lower. So that means the diversity is less. And what this interesting data, when it was further looked at, what we found is that the Collinsella taxa was much higher in abundance compared to the rest of the taxa. So further analysis shows that in those who were having NASH, in contrast to those who had healthy and obese, this Collinsella aerofaciens was significantly higher in abundance, possibly raising to, again, data suggesting, could this be one possible taxa playing a role in the Indian NAFLD and NASH? Again, the same data being suggested is that this was further the abundance was looked on the PCR, again suggesting that this Collinsella aerofaciens could be a possible reason, and the numbers were definitely statistically significant. So what is this bacteria? This is how it looks like, very tiny ones. And then what the fermentation products of leads to is production of ethanol, formate, hydrogen, short chain fatty acids, and lactates. So I'll not take much time. And this is again the Collinsella aerofaciens has been, the whole genome sequences has already been done by Dr. Bhabatosh and his team. So, and what we highlighted out here and what this study results suggest is that possible there are some differences in the Collinsella aerofaciens in patients with non-alcoholic steatohepatitis, which leads to is less metabolism of ethanol, leading to more accumulation. That could be one possible reason. But the other question what we raised is, how does this Collinsella aerofaciens is really playing a role? So this was looked in as a mice model where 4 different groups were taken. Normal diet, this was one which was CDHF diet, and the third group was normal diet with Collinsella aerofaciens taken from the NASH patients. And the fourth group was the CDHF diet with Collinsella aerofaciens taken from the NASH patients. So what the results suggest, in terms of fat, there was no difference in the groups, possibly suggesting that Collinsella aerofaciens probably is not leading to much of steatosis. But what it importantly showed is that probably it plays a role in causing more inflammation, as shown in this slide, where we see the pro-inflammatory cytokines, TNF-alpha, IL-6 were much higher in the CDHF with Collinsella group compared to the rest of the groups, possibly giving us some hints that this bacteria is probably leading to more inflammation, and that may possibly be playing a role. So with this, the take-home messages from my talk are, NAFLD is the most common chronic liver disease globally, and the burden is increasing, and it's leading to more chronic liver disease and advanced liver disease, and it is now becoming, or it has now become the most common indication for liver transplantation in the West, and we are not far behind. Patients with non-alcoholic steatohepatitis have lower gut microbiome diversity and richness compared to those healthy individuals and those who do not have NASH. There is an increased abundance of Collinsella aerofaciens in the gut of Indian patients, and which appears to be quite unique. And it is not the steatosis which this bacteria causes, and possibly it's more of inflammation. I think this is what was derived from this study which I had presented. And I would just like to end the talk over here again, giving the things back. So if you got any questions, please address to Dr. Bhabatosh. On the gut microbiome part, I'll be happy to take clinical questions. Thank you.
Thank you, Shalimar. Please join us here. We will move on to the third speaker, Professor Saurabh Datta, who is from the Department of Pediatric Medicine at the Postgraduate Institute of Education and Medical Research, Chandigarh. He is also an adjunct professor at the Department of Pediatric Medicine, McMaster University in Canada. He's got a PhD. He's one of the leading neonatologists of the country and has a PhD in some very interesting work that he's done in the young babies. So he's truly a physician scientist with a lot of work on premature retinopathy of prematurity, neonatal sepsis, and probiotics. Today we have him. Hi, Saurabh. Welcome.
Hello.
We have you presenting the breast milk microbiome with special reference to Indian mothers, where you have some interesting work to share with us. Welcome, and over to you.
Thank you, Dr. Shinjini Bhatnagar, for that kind introduction. And thank you, Dr. Bhabatosh, for having invited me to participate in this very interesting conference. Am I clearly audible, and is my first slide visible to all of you? Hello?
Yeah, you're audible. Please go ahead.
Okay, thank you. So over the next 20 minutes, I'll be delving a little bit about the breast milk microbiome with special reference to Indian lactating mothers. So the overview of my talk is going to be what is the source of bacteria that colonize the neonatal gut, and what role does breast milk play in it? What is the source of bacteria in breast milk microbiota in various countries with special reference to India, the factors affecting breast milk microbiota, and potentially pathogenic bacteria and antibiotic resistance genes that are present in preterm breast milk. So it is a complex topic, so I'm going to start off with the definition of a baby. So a baby is essentially an alimentary canal that is very demanding at the upper end and has no sense of responsibility at the lower end. So since an alimentary canal plays such an important role in the life of a baby, let us first try to answer this question. From where do the first bacteria get into the neonatal gut? So this was a study done way back in 1975 by Carlson et al. So what they did was a group of investigators went into the delivery room. They swabbed the baby's mouth in the delivery room, and they also took mother's rectal and vaginal swabs at the same time, and they cultured it for lactobacillus species. Remember, this was an era long before metagenomics and molecular identification of bacteria. So they cultured for lactobacillus species. And what they found was that there was a high correlation between the oral cultures and the rectal and vaginal cultures at birth as far as lactobacillus species was concerned. And they concluded that the vaginal lactobacillus species was getting into the baby's mouth during the process of birth, and the source of the vaginal lactobacilli was the mother's own intestine. On day 6, they noticed something interesting, that the oral cultures for these lactobacilli had become negative, but the neonatal stool cultures were now showing the same organisms. So they concluded that the first inoculum into the baby's mouth and intestines was probably from the maternal vagina and the perineal area. The question then asked was that, is that the only source, or is the breast milk also a source of bacteria for the neonatal gut? So this was a landmark study done in 2003 by a very famous group based out of Madrid, Rodriguez et al., published in the Journal of Pediatrics. They took 8 mother-infant pairs. They did cultures as well as some molecular tests of breast milk collected aseptically. So these are all the measures they took to ensure that the breast milk was collected aseptically. They also took swabs from the breast skin, the vaginal swab, neonatal feces, and the neonatal oral swab after a feed. And what they found was something which was revolutionary for that time. They discovered that the breast milk is not sterile. In fact, it's teeming with bacteria. It has 10⁻³ to 10⁻⁴ colony forming units per ml of milk. And there was a high concordance, identical RAPD profiles of the neonatal feces and the oral swab with the breast milk. And there was no concordance with the flora of the breast and vagina. So they concluded that the breast dominates gut. So the question therefore was, how do the bacteria get into the breast milk? How do the bacteria get into the breast milk? So there were 3 possible routes that were hypothesized. This was a paper published in 2022, which had summarized the evidence till then. Was it from the infant's oral cavity? That means the infant's oral cavity was getting colonized by the process of birth, and then that was causing reverse colonization into the breast milk. Or was it simply from the maternal skin getting through the lactiferous ducts, etc., into the mammary glands? And the third, which was a very unusual hypothesis, was it coming directly from the maternal GI tract through some kind of a hematogenous route into the mammary glands? So what was the supporting evidence for the infant oral cavity? Some people had detected a few oral bacteria in the, and the alleged mechanism was that there was a retrograde flow of milk from the infant's oral cavity to the mammary ducts. For the maternal skin, a few human skin commensals had been reported on and off, and the mechanism was thought to be colonization of the mammary gland by maternal skin microbiome through the nipple. Maternal GI tract, the supporting evidence was that there was an abundance of what are strict GI anaerobes, normally not found anywhere else. For example, bifidobacterial species, Bacteroides, Clostridium, they were found in abundance in human milk. And more about the alleged mechanism in the next slides. So the question therefore was, which is a more important source? Is it the vaginal flora, or is it the the breast milk as a colonizer of the neonatal gut? And does breast milk get reverse colonized from the baby's mouth, or is the mechanism? To answer this question, Rodrigues published this elegant study in 2007, where they used 16S rRNA gene-based PCR. They collected breast milk aseptically from 3 sources, and they compared this with each other. So 5 mothers had an elective cesarean section with no rupture of membranes. So that means there was no question of bacteria having ascended through the ruptured membranes into the amniotic sac and got into the baby. So these were elective cesarean sections. 5 mothers with vaginal delivery. That means the babies went through the birth canal, and they collected pre-colostrum even before the baby was born. So there was no question in the third group of bacteria going from the infant's mouth into the breast milk. They extracted bacterial DNA, and they did the following things with the DNA. And what they found was very surprising. One, there was no difference in the breast milk microbiota, no gross difference between the cesarean born babies and the vaginally delivered babies. So that means the ascending bacteria, or the bacteria acquired by the baby during the process of delivery in its mouth, had no connection with the breast milk microbiota, because even cesarean born babies had it. The breast milk was replete with bacteria. This was reconfirmed. And these organisms were present even in the pre-colostrum before delivery. So they concluded that reverse colonized breast milk was not an important source of bacteria in the breast milk, and the breast milk is an important source of bacteria for colonizing the neonatal gut, and it is probably more important than the vaginal flora. The question, therefore, is how can bacteria from the mother's gut get into the breast milk? So if it is not reverse colonization, if it is not from the skin flora, it must be coming from the mother's gut, but how into the breast milk? So the hypothesis was that there is a gut-mammary axis. So we do know that immune cells from the gut, Peyer's patches and lamina propria, do home in on the mammary gland in late pregnancy and lactation, and they do get antibodies from the Peyer's patches to the mammary glands. So the hypothesis was that probably similarly area from the maternal gut. So, in a strange way, the mother's gut and the baby's gut are kind of intertwined. The bacteria from the mother's gut are getting into the breast milk, from that to the baby's mouth, and from there into the baby's intestines. So the evidence for the gut-mammary axis came from this landmark published in 2007 by Perez et al. So what they did was they took human mother-infant pairs, and they performed various molecular studies on maternal feces, maternal blood, maternal milk, and then also from the infant's feces, and these were all breastfed infants. And what they found was really created a paradigm shift in our thinking, because they found that there were identical molecular signatures of a bacteria in the maternal feces, maternal blood, maternal milk, and the infant feces, thus closing the loop. So they also visualized bacteria inside milk monocytes and peripheral blood monocytes, identical bacteria. And they went ahead and did animal studies. So mice models were created. This shows at the bottom control mice, pregnant mice, and lactating mice. The black bars are showing bacteria in mesenteric lymph nodes. The gray bars are showing bacteria in the mammary glands of these mice, and see what happens. Control mice, there are hardly any bacteria either in the mesenteric lymph nodes and virtually none in the mammary glands. Pregnant mice, there's a huge number of bacteria which suddenly appear in the mesenteric lymph nodes, which are draining the gut. And lactating mice, those bacteria are now less in the mesenteric lymph nodes, and they have now found in the mammary glands of these mice. So putting it all together, it looks like the bacteria are being carried by some specialized immune cells from the mother's gut to the mother's mammary glands. And that is happening precisely at the end of pregnancy and lactation, when these bacteria are going to be needed the most during breastfeeding in order to colonize the neonate's gut. So which are the cells that are carrying these bacteria? The main hypothesis, which is widely accepted now, is that these are dendritic cells. So this is a cartoon picked up from Nature Immunology, which shows this dendritic cell out here. On the top is the gut lumen, which is showing all these bacteria. So the dendritic cell is somehow going and sampling these bacteria and picking up only the ones that are required for the baby. There must be some highly sophisticated mechanisms, maybe some molecular patterns being expressed on the surface of these bacteria, which is being recognized by these dendritic cells. They are incorporated into the dendritic cell, and the dendritic cell is then transporting it by some mechanism. And maybe it is not the dendritic cell which is going all the way to the mammary gland, but it is handing it over to some other specialized cells, probably of the monocyte macrophage lineage, which is carrying these bacteria through the bloodstream and lymphatics into the mammary gland, and then during breastfeeding into the baby. So seeing is believing, and this was an electron micrograph showing dendritic cells which express tight junction proteins and extend the dendrites to sample bacteria. Actually, been shown to sample bacteria from the gut lumen without disturbing the gut brush border epithelial cells. So it's kind of squeezing between two epithelial cells, picking up bacteria from the intestinal lumen and carrying them. This is another electron micrograph showing that dendritic cells are engulfing lactobacillus cells and transporting them across CACO2, which is basically a colonic cell monolayer in a petri dish in a laboratory situation. So here, dendritic cells actually, engulfing bacterial cells and then being shown to carry it across to the other side of the layer. Changing country of origin affect the position of the milk microbiota, or is it something that is relatively constant all over the world? So to answer this question, Kumar et al. From Finland conducted this study, which they published in 2016. They took 10 healthy, exclusively breastfeeding countries. They analyzed the V4 region of the bacterial 16S rRNA gene, and they did 16S RNA gene metagenomics. The 4 countries were Spain, China, Finland, and South Africa, which is being shown at the bottom. And they show the relative abundance of various key phyla in this chart. So there are 4 key phyla that looked at: Proteobacteria, Firmicutes, Bacteroides, and Actinobacteria. And this is the relative abundance expressed as percentage. Now note that there are major differences between countries. The first thing that strikes you is that in South African mothers, the Proteobacteria is the most abundant phylum. And that doesn't seem to be the case amongst Caucasian and East Asian mothers. Secondly, Bacteroides, the gray part, was far common amongst Spanish mothers compared to mothers from any other part of the world. Thirdly, Actinobacteria, mothers from China, had a far greater preponderance of Actinobacteria, the maroon part of the bar, compared to mothers from other parts of the country. So it appears that the bacterial composition of breast milk is not something which is constant across the globe. So there are differences even within countries. So, within large countries, there could be differences in different parts of the country, and that may well hold true even for India. So this is a study from China where they took Taiwan and 6 regions of China, and they did metagenomics from the V1, V2 region of the bacterial 16S rRNA gene. These pie diagrams show the relative, abundance of different bacterial families from different parts of China. And at a glance, you can make out that these big pies, the blue and the orange sectors of these of the pie chart, they show relatively different abundance in different parts of China. just emphasizing that even within a country, regional, maybe dietary, maybe climatic, or whatever differences, we don't fully understand what the reasons behind these differences, but there are major differences from different parts of even within a country. And the other interesting thing is, and we'll be coming to this later, look at the orange sectors, Pseudomonaceae. That means
Saurabh,
the
you
bacterial.
would have another 5 minutes because
Okay.
yeah, sorry to interrupt.
So there are certain regions that are dominant in Pseudomonas genus. So are Indian mothers any different? So this is a study that we did in collaboration with Dr. Bhavatosh Dash. We took 20 breastfeeding healthy mothers with no recent antibiotics, and we extracted the DNA, and we looked at the V1 to V5 regions of the 16S RNA gene. So these were already all healthy lactating mothers. And what we found was that Proteobacteria was overwhelmingly the dominant phyla. When we looked at the major genera, Pseudomonas was the major genus that we seem to be dealing with. And we looked at the individual species. These were the species. We looked at each individual species. None of them is a known pathogen, although the word Pseudomonas seems to conjures up, images of a dangerous thing, but none of these are actually pathogenic. These are all commensal Pseudomonas species. When we clustered subjects based on the relative abundance of Pseudomonas in the Shannon index, we had 3 subgroups which seem to appear: those with high Pseudomonas, low diversity, low Pseudomonas, and high Pseudomonas, high diversity. And this was the heat map again showing the same. Our data seem to match one previous study that had been published from India by Vaidya et al., which they again did 16S RNA gene, where so the left-hand side is the study from Vaidya et al., where they too had a very high preponderance of Proteobacteria compared to other studies from Caucasian mothers. So Vaidya et al.'s data resembled ours. And amongst their relative abundance of the top 10 genera, they too found Pseudomonas to be a very important genus. So this study, the weighted unifrac distance comparing Indian, Finnish, and Swiss mothers, showed that the composition in India is very different from that in Caucasian mothers. This is another study again showing that from Indian mothers, Proteobacteria is a dominant phylum. So Pseudomonas in human milk, could it be pathogenic? Could it colonize the neonatal gut? And is it commonly reported? Well, as I said, no Pseudomonas species detected in our study was a known pathogen. Some of these have been known to cause sepsis in highly immunocompromised adults or when contaminated IV fluids given. Just a few reports in adults. And all the neonates in our study were clinically normal on 1 month of follow-up. And no infection due to Pseudomonas from human milk has been reported so far in literature. This is again showing the same thing, that Pseudomonas hardly ever actually colonizes the neonatal gut to cause infection. So why is there so much of variation in human milk microbiome? So this diagram kind of summarizes the reasons behind this diversity. All previous studies have used 16S RNA gene metagenomics. We are currently doing a study using whole shotgun metagenomics to study the factors affecting the human milk microbiome. This is still work in progress. We are going to be looking at 40 mothers delivering preterm and looking at various things like BMI, exposure to antibiotics, vegetarian versus non-vegetarian diet. And this is a PhD student of mine who's doing this. And just quickly going over what she has found so far.
Yeah, may want to.
I think I can wind up because I've covered the most essential aspects of what I had to say. And I'm happy to take any questions after this. Should I stop sharing? I can stop sharing.
Yeah. Because we have about 20 minutes for discussion. And so thank you all the speakers. Very interesting insights. We'd like the audience now for their feedback and any questions. Yes, please.
So Dr. Shalimar, if I can start with, okay. Yes, please start.
Yeah, my question is to Dr. Yafi. When you introduced antibiotic, ciprofloxacin, and studied for maybe a few months. I forgot the exact time scale. And you showed there is one A to G mutation and in the alignment. I also noticed there is a short deletion. Is this a sequencing artifact or is this coming frequently and it is actually getting deleted?
Yes, but first of all, the Cipro was introduced for just 5 days. We were just tracking for many days. And that specific slide is just an illustration. That's not real data. We did follow substitutions and insertions and deletions, and we can see that substitutions play a much major role compared to insertions and deletions in general, but we're tracking them as well.
Good morning. My question is to Dr. Salimar. Can fatty liver disease be reversed using fecal microbiota transplant or by changing the lifestyle?
So I think you asked a very important question regarding the fecal microbiome transplant. At present, there is no available data to suggest. This is being looked at in studies. And I think over the next couple of years, we'll have some idea whether fecal microbiome transplantation could be of some use and in which subgroup would it be beneficial. So at present, it's mainly in the study stage. Your next question is lifestyle. Yes, lifestyle is the most important part of the management. So when we manage fatty liver disease, you take care of the diet, you take care of the exercise. These are the two things which are available right now. There are no approved drugs, but there are a lot of drugs in pipeline which are being again studied. So we'll have those results in the coming years, how it really benefits. So yes, lifestyle is the major backbone of treatment of fatty liver disease at present.
Thank you, sir. And one more question to Dr. Datta. You said that bacteria from the mother can come into the newborn, and it has also been observed that AMR bacteria are also present in the neonatal kids just after the birth, within few hours. So is it possible that the same dendritic cell is playing role in taking the pathogenic bacteria into the newborn child through breastfeeding?
Yeah, thank you for that question. In fact, that was going to be covered in some of the slides that I skipped because of shortage of time. Yes, I think that is very much a possibility that multi-drug resistant bacteria and antibiotic resistance genes are being transported from the mother's gut to the mammary glands and from that into the baby's mouth and into the baby's gut. So in fact, we had some data about that showing a high prevalence of antibiotic resistance genes in the baby's mouth and in the in the breast milk, and a fairly high concordance of the kinds of genes that we are seeing between the two. So it's quite possible that these must be hitchhiking on that same pathway, the dendritic cells and possibly the mononuclear cells thereafter, to find its way into the neonate, which just shows that rampant use of bacteria in the mother can ultimately affect the health of her neonate.
My question is to Dr. Yaffe. So most of us probably take antibiotics at least once a year, I believe. So would you think that we are constantly really accumulating resistant bacteria, and what is the consequence of this accumulation? I mean, at some point of time, should we all be worried that we may get some infection with commensal bacteria that are antibiotic resistant?
Thank you for that question. So the whole study is regarding commensals. So on the face of it, it doesn't really matter if your commensals are developing resistance. It actually might be a good thing. But when you think about the pathogens that might be under the radar, they might affect the general population. So not necessarily you. You're just, if you carry a pathogen and that pathogen becomes resistant, then it can move further. So I think it's more a question of policy than a question of any specific person. But one thing that's very interesting that comes out of the study, that specifically for Cipro, the size of the population is very important. So one might think of a clinician, a future clinician assessing and deciding to give Cipro only if a pathogen has a small enough population, and if the population is larger and susceptible, then they might actually decide not to do that, because they know that the gyre mutation might occur. Every antibiotic is obviously different, so this study needs to be repeated again and again. But that's, I think, the gist of it. I would say is that I wouldn't change anything right now, but these are ways to guide future decisions.
So there is no evidence of any drug-resistant commensal bacteria gaining some virulence and becoming able to infect people. There is no such evidence, even in animal models, for example.
I actually don't know.
Nishit?
Can I ask one question to Dr. Shalimar after?
Okay, maybe we'll come back to you, Guru.
Yeah. So, Dr. Shalimar, is there any association between fatty liver and propensity for viral infections? Do people with fatty liver are more prone to, let's say, viral hepatitis, for example?
So I think that's a very good question. So per se, viral hepatitis, no, but let's look at what data we got from COVID. So there's now a lot of data available in terms of predictions of severity and outcomes. So patients, those who got fatty liver and those who got obesity, they had higher mortality. So it is not its predisposition, but it's the outcomes may probably be affected by the underlying obesity and associated metabolic parameters in those. So that group may have a more severe presentation and possibly a higher risk of mortality. That's what we know. But for simple viral hepatitis, there is evidence to suggest that those who got fatty liver disease may have more severe disease. Whether they get more frequently, that's not very clear. But then they get more severe presentations, and outcomes are because fatty liver disease per se also causes some amount of fibrosis in the liver. So your regeneration capacity over there is low compared to a healthy individual. So therefore, possibly the outcomes are poor in that individuals when they get a viral infection.
So my question is to Dr. Shalimar. I'm Nishit from THSTI. So you have shown that Collinsella leads to inflammation and not the active disease. So can we believe that any other bacteria, say E. coli, will also cause the inflammation? That is my question number one. So how specific that phenotype is as far as it is relating to the NAFLD? And secondly, in the mice model, usually we also see in the humans that the active disease takes longer period of time from various grades. So do we expect that if the same length of time were to be used for mice, we would have seen some sign of active disease in them also?
So I think for the first question, I have my lifeline over there, Dr. Bhabatosh, who's going to answer it. The second thing is, like, say, there is a difference in the mice and human in terms of lifespan. This is a mice model. There are lots of issues with animal models when you extrapolate to humans. You don't see that whole spectrum. In humans, it's a slow disease. In fatty liver disease, I'm looking at not even years, decades. So anything to happen move from one stage of fibrosis, you're looking at an average of 7 to 10 years. It's a slow moving disease. So it takes time. And our understanding is evolving on that. But regarding do E. coli or other microbiota play a role, I think Bhabatosh can give an answer from his experiments.
Come back to you.
We will discuss later.
Yeah. So I have a.
We'll come back. Bhabatosh wants to respond to Nishit's question, then we'll come back to you, sir.
So I think inflammation is not any bacteria specific. Okay, so any bacteria, if they have the inflammatory molecules, they can induce the inflammation. In Collinsella, what we observe that molecule they produce that is quite unique. If you look at the E. coli induce inflammation through LPS. But what we observe that Collinsella, they have a specific signature that produce excess ethanol. And ethanol is one of the strong inflammatory molecules. So in Indian context, we find the Collinsella, but if you look the other literature in the China, they found that there is a Klebsiella. And they do also reported that Klebsiella have accumulated these ethanol production functions. There are other studies, they do also publish that it could be the Candida and also sometimes the Saccharomyces. It's not the microbe, the important is the functions. If you have this function from any microbe, they can induce inflammation and cause the disease.
So can the source display whether it will lead to NFL?
Not getting
So of information like ethanol, triggers will decide whether the NFLD will be progressed or not.
So NFLD is a multifactorial disease. It's not only the one. Only inflammation may not lead, but if you have the additional risk factor, definitely they can induce the disease progression. Sometimes also the disease causes.
Okay, thanks.
So I have a question for Dr. Eaton. So you talked about the parameter of evolvability. The parameter of evolvability. Yeah. So you talked about the two parameters that you were considering, and I was wondering if there is any way to factor in or model a factor where the exposure to the drug because of the colony structure can be factored in, because based on the colonization structure above the mucus layer, the amount of exposure of the antibiotic to the bacteria would be different. And is this something you can model or factor in some way?
Thank you. That's a beautiful question. So, this is a very simple experiment, which we do not know anything about these commensals and where they reside in the gut. But that's the fact that we see difference between different species and their evolvability attests. We know that there is something to do with the way they are exposed to the drug, either the way they create biofilm and they reduce their exposure or their spatial composition, so many things. So we cannot add that to the model because we don't have any measure. I only use simple measure that I have, but definitely that would be a very valid hypothesis for what is driving differences between different species.
Thank you. And I have another one for Dr. Shalimar. So in one of your slides, you talked about the feeding the mice with the CDHF diet. If I don't know if I'm actually having the name right of the diet, the second diet that you had along with the normal diet. And there was some sort of inflammation that was also showing up only with the CDHF diet. And this sort of amplified when you add the bacteria to it. So do you think it is amplification, already inflammation that is happening is sort of amplified by the bacteria, or is it the other way around?
So I think one was the chow diet, that was a CDHF diet. CDHF diet, we're looking at the steatosis which is occurring. Yeah. Now, whether that per se leads to more inflammation being happening or per se causing that, I think Babatosh may give an answer to that with his understanding. Does it really, which is existing there, it causes increased, or it is per se the only soul? But I think it's not the one bacteria alone, it's multifactorial. So I think that still is not clear.
Thank you.
There was a question.
I have a question to Saurabh. As you suggested that the dendritic cells there who are picking up the antigens and carrying them back to the brain. So gut very conducive around.
I'm sorry, I can't hear.
Yeah, he's going to ask again.
Yeah, the dendritic in the gut lumen is not a very conducive environment for the cells to survive. So how do you think that the dendritic cells should be reaching there and picking up the antigen? And then once the dendritic cell picks up an antigen, it gets activated. And then when it carries the antigen load to the breast, it does not trigger any inflammatory response?
I think many of these questions have not yet been answered satisfactorily in the medical literature. In fact, there are a few of these very questions that we are trying to address in some ongoing studies. So these dendritic cells are found in the lamina propria and the Peyer's patches. They are not found in the lumen. So it is from outside the lumen, from the wall of the intestine. They're probably extending their processes between epithelial cells, sampling these bacteria and bringing them back into the cell. The dendritic cells are probably not traversing the entire path from the gut to the mammary gland, because they don't normally do this kind of a long distance traveling. And it's quite possible that, these bacterial cells are actually being handed over to some other specialized cell of the monocyte macrophage lineage, which is finally homing in on the mammary gland through some sophisticated, cell recognition method using chemokines and so on. My hypothesis is that, and although nobody's ever proved it, that within the mammary gland, these bacteria are probably being held intracellularly within the macrophages, and therefore they are being shielded from the both from the lactose, which they are supposed to be fermenting, because they don't ferment it in the breast milk itself, because these lactic acid bacteria and lactose at an ideal body temperature, 37 degrees centigrade, are present in the breast milk, but it is not fermenting the lactose. So these cells are probably being held in some sort of an intracellular location in the mammary gland, being protected both from the immune system as well as from the lactose. But there is no proof for any of this.
My other question is to Dr. Shalimar. When we look at your inflammation data, you have shown the activation of NF-kappa B, TNF-alpha, IL-6, and IL-10. Barring TNF-alpha, like IL-10 is an anti-inflammatory cytokine, and IL-6 plays both pro and anti-inflammatory roles. So based on this data, maybe this is a preliminary data, how can we say that inflammation is responsible for all the pathophysiology associated?
So we were just trying to show what happens to these cytokine levels. IL-10 was low, the TNF-alpha and IL-6 were high. So this is preliminary data, and I agree with that. But then this suggests that the rise in this pro-inflammatory in contrast to a reduction in anti-inflammatory that's what we are hypothesizing over here. That's what the results are. So.
Thank you.
Yeah, first the lady here and then.
Dr. Shanta from NICED. So my question is to Dr. Shalimar. Like what percentage, you have shown that candida, that the fungus is also responsible to cause change in the microbiome, and it may lead to NASH. So what percentage of NASH is reversible? Like with this food and all lifestyle, like if NASH affects the liver almost more than 90%, then can we say that it is reversible with following all these lifestyle and diet?
So let me give you the global picture. You got 100 fatty liver disease, 20 of them would have NASH or MASH. Okay, so it's a small fraction. Within this group, when you take care of the diet and lifestyle, that means you're effectively causing a weight loss. A reduction in weight loss, say somewhere around 5 to 7% of the body weight, would lead to a resolution of NASH. So this data is supported more by the bariatric surgery data. So bariatric surgery is the surgery which we do in individuals who are very obese, and they undergo a surgery in which the stomach is reduced in volume to lead to weight loss. Over there are studies in which people have done paired liver biopsies. That means liver biopsy today documenting the patient has NASH, and then you do it 1 year down the line or 3 year down the line, 5 year down the line to see what is happening. There, the resolution happens in NASH in almost like 90% of patients when you lose weight. So lifestyle changes are associated with resolution of NASH. In a subset of patients, even fibrosis can also be reversed. So it is reversible. We need to take adequate steps for taking care of the disease.
Yeah. Thank you. And one more question to Dr.
I think you should, we'll just come back to you, ma'am. He's waiting.
So my question is to Dr. Datta. So very naive question. So what is the association between the microflora of neonatal infants, neonates, as well as the amino fluids?
I couldn't catch it. Which fluids? Sorry. Yeah.
The question is the association between amniotic fluid versus neonates. Because I'm not hearing any information including existing literature?
Okay, yeah. So that's an interesting question. There was a time when everybody was doing this metagenomics of all possible fluids, including from the fetal gut, the placenta, the amniotic fluid, and so on. And the initial data that came was that none of these are sterile. They were earlier thought to be, we were always taught that these were sterile fluids, but then we went through a phase where it was shown that the placenta has a microbiome, it is not sterile, that the amniotic fluid is also not sterile, and that the fetal gut is also not sterile, that the fetal gut, the neonate keeps swallowing amniotic fluid during in utero life, and probably the amniotic fluid is a source for colonization of the fetal gut. However, having said that, of late, some recent studies have actually challenged this viewpoint and have shown data to suggest that probably the previous studies were flawed because of contamination. That despite whatever precautions they had taken, the placental samples had probably got contaminated, because it's very difficult to prevent contamination of placental tissue. No matter how carefully you collect it, there is always a danger of, and these methods are so sensitive that it's very easy to pick up a contaminant. So there are broadly some two big studies that have shown that the placenta does not have a microbiome, and there are equal number of studies that are showing that it has a microbiome. And this is a very contentious issue right now. So similarly, it holds true for the amniotic fluid. So I don't think we have the last word on it, whether the amniotic fluid has an independent microbiome like the placenta, and whether that's causing the fetal gut to already have bacteria even before the baby is born. So this is still an open question. Anyway, thank you for bringing up this issue.
May I ask?
Maybe we can go for the last question.
Yeah. So this is, my question is to Dr. Datta. So this is because you have said that even baby born with LUCS will have the microbiome since the first day of life from mother's breast milk. So now it is a difficult then to get rid of this problem. So is there any hypothesis how we can prevent that?
No, it is not a problem. It is desirable. See, the breast milk is supposed to have a microbiome, a very diverse microbiome, which is responsible for colonizing the neonatal gut. So these are commensal bacteria, mostly bifidobacteria and other bacteria, which are required for the normal colonization of the neonatal gut. The neonatal gut is supposed to get colonized.
I understand that, but you have said resistant bacteria also gets colonized.
So that brings us to the larger question of overusage and misuse of antibiotics. Ultimately it boils down to that. So what happens is often any mother who comes with threatened preterm onset of labor, who has a premature rupture of membranes, is virtually bombarded with antibiotics. And especially those who deliver preterm, they end up sometimes receiving several courses of antibiotics. Now, those antibiotics are coming at a price, because they are selecting out resistant bacteria, antibiotic resistant genes in the mother's gut. They may also be selecting out resistant bacteria de novo in the bacteria which are already there in the breast milk. So, a certain proportion of bacteria will always be harboring antibiotic resistance genes in any population, but they get selected out. And they are now finding their way into the neonate's gut because of breastfeeding. So, if one really wants to tackle it at its source, one has to have antibiotic stewardship policies that restrict the use of broad spectrum antibiotics in the mother, especially those delivering preterm.
Okay, with this last interaction session, we close the session. And on the behalf of all the here chairs, I would like to thank Dr. Saurav Datta, Dr. Shalimar, and Dr. Yatan for the wonderful seminars, and the audience for wonderful questions to make this session highly interactive. So with that, I will hand over to you.
Now I request, thank you to all the session chairs and the speakers for the exciting and informative session. Now I request session chairs to kindly felicitate the speakers. I request Dr. Shinjini Bhatnagar to hand over the token of appreciation to Dr. Eaton Yap. Dr. Avinash Bajaj to hand over the memento to Dr. Shalimar. Now I humbly request Dr. Shibnath Majumdar to kindly come up on the stage to felicitate the session chairs. Ma'am, please. To Dr. Shinjini Bhatnagar. Dr. Avinash Bajaj. And Dr. Ganesan Kartikeyan. Thank you, sir. Thank you, ma'am. I request all the speakers and session chairs to join for group photo. So now we break for tea. I request the conference volunteers to assist the dignitaries to the area of tea. We request the participants who are presenting the poster today be present near their posters. I request everyone to assemble back by 11:30 a.m. for the start of the next session. Thank you. So we'll start in another 5 minutes. Everybody, please be available in the auditorium. We'll start in another 5 minutes. Everybody, please be available in the auditorium. We'll start in 5 minutes. Everybody, please be available in the auditorium. We are about to start the session. Everybody, please come back to the auditorium. No, I'm not doing it. Sudarshana is doing I request everybody to come to the auditorium. We are just about to start. Circle. Welcome back, everyone. Hope you had fruitful discussions over the tea. Now we begin with the second session of the conference on vaginal microbiome and prediction of birth outcomes. I humbly call upon the stage the session chairs, Dr. Sudhanshu Vrati, Dr. Nitya Wadhwa, and Dr. Shibnath Majumdar. Ma'am, please. Sir, please. Dr. Sudhanshu Vrati, it is a pleasure to have with us Dr. Sudhanshu Vrati, who has been the former director of the Regional Center for Biotechnology, Faridabad, India. Dr. Vrati's lab focuses on key aspects of the JEV life cycle, like receptor binding and entry mechanisms, molecular mechanisms of virus replication, assembly, and egress. The lab has also been involved in the clinical development of an oral rotavirus vaccine for children. Professor Vrati is a J.C. Bose National Fellow and has several publications and patents to his credit. Thank you, sir, for joining us today. Dr. Nitya Wadhwa is a trained pediatrician who has transitioned to become a physician scientist. She has been doing clinical research for more than a decade and leads her team at THSTI. She has been associated with several large multicentric clinical trials, and she has been instrumental in establishing a large longitudinal cohort for pregnant women with the primary aim of building risk stratification algorithms for adverse birth outcomes. Thank you, ma'am, for joining us today. Dr. Shibnath Majumdar is a professor and dean of Faculty of Life Sciences and Biotechnology, South Asian University, New Delhi. Professor Majumdar's team worked towards unraveling the various pathogenic mechanisms of zoonotic pathogens and to develop intervention strategies against them. He has established the stellar role of virulence plasmids in Aeromonas hydrophila pathogenesis and successfully used plasmid-cured isolates as vaccine candidates against its spread. Thank you, sir, for accepting the invitation. In this session, each talk will be of 15 minutes. After the talk of all the speakers, there will be a 10-minute long question and answer session. I request the audience to kindly preserve their questions till the end of the session. One of our speakers, Dr. Jeffrey Klausner, will be joining us online. I now request the session chairs to introduce the speakers and call them over for the talks.
Good morning, everybody. Welcome back to session 2 on second day, which is on vaginal microbiome and prediction of birth outcomes. I'm very happy to introduce the first speaker, Dr. Gregory Buck. He's a professor at the Department of Microbiology and Immunology at Virginia Commonwealth University. He deals mainly with the area of human microbiome project, narrowing it down to issues related to bacterial vaginosis. He leads the Vaginal Microbiome Consortium, which is focused on developing an understanding of the consortium of the microbiome of the female reproductive tract on women's gynecologic and reproductive health. Dr. Buck is going to speak on microbiome of the female reproductive tract and women's health. Dr. Buck, please.
So thank you for the wonderful introduction. And thank you to the organizers for inviting me to come to this wonderful conference here in India. This is my first visit into India, and I've enjoyed it so far very much. Not only just seeing the Taj Mahal, but also interacting with all the talented investigators that I've met here. And I look forward to continuing these discussions and setting up hopefully collaborations with people here in India. So yeah, I'm going to talk about some of the work we've done over the last few years on the vaginal microbiome, or microbiome, and try to give you some kind of a flavor of what we've been doing in our work. I may be preaching to the choir here, but why study the vaginal microbiome? Well, it's a clear but still pretty much undefined, has a role in women's gynecological and obstetric health. Bacterial vaginosis itself is still an enigma. It affects a high percentage of women worldwide, ranging in from 18 to 40 or so percent. It's recurrent. 50% of the bacterial vaginosis recurs within 6 to 12 months. We don't understand that etiology. And women with bacterial vaginosis are at risk of higher risk of being infected by STIs, viral infections, et cetera. There's certainly an impact on pregnancy and outcomes at birth. And that's been one of our objectives, to look at the impact of the vaginal microbiome on adverse outcomes in pregnancy. So why are we interested in preterm birth? Again, this is probably preaching to the choir here, but it's not a trivial problem. At least 10% of pregnancies end preterm worldwide. In some places, those numbers are much higher. Low and middle income countries sometimes are up to 20, 30% preterm. And the preterm, as you're probably aware, has a lot of effect on the outcomes of acute outcomes right after pregnancy and long-term physiological outcomes, including immune diseases and intellectual debilitation. So it's a real important problem, and there's increasing evidence that the vaginal microbiome, the microbiome of the female reproductive tract, plays a role. So I'm going to talk to you about primarily data coming from two major studies. There were studies funded by the NIH Human Microbiome Project. We were fortunate to be funded twice there. In the first project, we were looking at cross-sectional study of around 4,000 women. I think we had 6,000 visits, looking at the microbiome of the female reproductive tract and trying to correlate it with outcomes of women's health. In the second project, it was more focused. It was a multi-omic project where we were looking at pregnancy. I think we had about 1,500 pregnancies, and we're looking at the impact of the microbiome and other omic approaches to the impact of that on preterm birth. So I'll talk to you a little bit about that today. So the one thing about the vaginal microbiome is that it's considered to be a homogeneous microbiome. In other words, the dogma is that the healthy vaginal microbiome is primarily occupied by, colonized by species of lactobacillus. And that's kind of contrary to ecological principles. It's generally thought that a more complex population or ecosystems like you'd see in a coral reef, for example, is more healthy than, say, a wheat field. So a particular disease could get in and destroy a whole wheat field, whereas in a complex ecosystem, a individual impact would not have that kind of an outcome. So it is considered that if the vaginal microbiome really is homogeneous, that is a kind of anti-ecological situation. And if this actually has been tested. This is a stack bar chart looking at the vaginal microbiome of around 143 young women that were part of the HMP Jumpstart project about 10 or 12 years ago. And it's a genus level stack bar chart showing lactobacillus in yellow and everything else in different colors. And what you can see here is that largely all these women were colonized by some taxon of lactobacillus. This is probably a little bit of an oversimplification. This again on the left-hand side is showing a slide. So in the same slide of those same women, this is a genus level characterization. In the right-hand side on the top, the panel shows an analysis, a similar analysis of about 366 women in our vaginal human microbiome project, the first project that we looked at. And what you can see here is this is a genus level analysis, and these are in theory healthy women with no diagnostic outcomes or no obvious complaints. And what you can see is the microbiome is not as homogeneous as one might expect. And if you look at this at the species level, which is the panel below on the right, what you see is that the microbiome really is not all that homogeneous. It's not all dominated by a lactobacillus. There's a large percentage of lactobacillus. And in other words, there's about 50% of these women their microbiomes are primarily lactobacillus, but another 50% are not primarily lactobacillus. But what you can see is that in any individual woman, there is a high probability that their vaginal microbiome will be populated by a single taxon. And that is emphasized in the next couple of slides. Well, this is going back, stepping back a little bit. This is the first modern molecular characterization of the vaginal microbiomes done by Jacques Ravel and Larry Forney. It's a statistical analysis of the microbiomes of several hundred women. And what they were able to show from here is that the microbiome can be partitioned into what they called community state types. And they had initially 5 community state types, 4 which were associated with lactobacillus species and 1 that was more complex. More recently, they've had a higher resolution of the CSD4 so that now they have a more specific description. We've done that, but we've done some other things to try to look at the vaginal microbiome. We call them vagiotypes. So we say that if there's a predominant taxon present in the microbiome, we'll call that a vagiotype. So if there's a Lactobacillus crispidus vagiotype, Lactobacillus jenseni vagiotype, there's a Gardnerella vagiotype, a BVAB1 vagiotype. And they're actually, if you do the analysis like we did with very high level numbers of women and fairly deep analysis, you can see that there are around 20 vaginotypes that are represented in women where they are predominantly a single taxon. And those vaginotypes are just listed here. And you can see there's some major ones, some more prevalent ones, and then there's some less prevalent ones. But the take-home message from this particular slide is there's only around 10% of healthy women that have a vaginotype that's truly complex. There are women that have different vaginotypes with different taxa being predominant, but they're predominantly a particular taxon. And a very relatively small fraction of those women have what we would call a complex vaginotype, which has representation of many different bacterial species in them. So we looked at the question of one of the things that we're really interested in at my institution is racial disparities. We have a population in the area that's highly descendants of African descendants. We also have a fairly high population of Hispanics or Latinas in the area. So we wanted to look at, are there racial disparities? And what you can see from this slide, the top slide shows a stack bar charts looking at around 1900, or I guess it's 1700 women of African ancestry. And the bottom one is of around 400 women of European ancestry. And you can clearly see a difference here. We've done this in a number of different ways. This is not actually case matched, but we have done it case matched. And there is clearly a difference between women of European descendants and women of African descent in our population, in our cohort. And on the right-hand side, you can see the taxa that are affected, the ones that are more common in the African descendants and the ones that are more common in the European descendants. So there is clearly a racial bias here. And we're not really sure why there's a racial bias. It could be genetic, it could be customs, it could be environmental. We have done a genetic analysis that would identify a contribution to the human genome to the presence of Lactobacillus crispus in European women of European descendants. There was a tendency to look at BVAB1, or what's called lactic curva vaginae now, was not significantly associated with women of African descent, but probably it's there. We feel like there probably is a contribution. And we want to ask what happens to the vaginal microbiome during pregnancy. And this is just showing a comparison of some 1,000 of our vaginal microbiome cohort. Of around 1,000 women were pregnant, and maybe 3,000 women were not pregnant. So we compared those two, and we could just see here, again, this is not case matched. We did a case match study and it gave us the same result. There is a difference between the women that are pregnant and women that are not pregnant, and the women that are pregnant have a much higher percentage of lactobacillus dominated vaginotypes. On the right-hand side, there's a slide that shows that the effect is more obvious in women of African descent than it is of women of European descent. And that's likely because women of European descent generally start with more, before they're pregnant, start with a higher percentage of lactobacillus taxa in their vaginal microbiome. We then asked the question, when does the pregnancy change the vaginal microbiome? And we could see that during pregnancy, that in the first trimester, there was a relatively high proportion of non-lactobacillus vaginotypes. But as you progress into the second trimester, that number of non-lactobacillus vaginotypes decreased significantly. And the bulk of these changes happen early in the second trimester, early by the second trimester. The changes, again, for reasons that I guess I already said, probably seem to be more apparent in the women of African descent than in women of European descent. We think that this is probably driven by estrogen. We tested that indirectly in a way. We looked at the different contraceptive types, and we found that estrogen-based hormonal contraceptives, over-the-counter contraceptives, it reduced the diversity or increased the percentage of vaginal microbiomes that are dominated by lactobacillus species. You can see that here from the upper one is there's overall non-pregnant versus the women taking over-the-counter contraceptives. And the left on the right just shows the comparison between women who take over-the-counter contraceptives versus women who use condoms. And there is a clear preference of lactobacillus species in women who use over-the-counter contraceptives. And then there's there's a lot of bacteria that are considered less optimal for women that were associated with use of condoms. So we asked the question, does the vaginal microbiome influence the risk of adverse pregnancy outcomes? That was using the data from our mom's PI study, the second HMP, NIH HMP funded study that we did. In this study, we looked at around 1,500 women longitudinal through pregnancy, taking quite a few different swabs or different samples from different sites, mostly focusing on the reproductive tract, but also we took buccal, rectal, and skin samples, which I'm not going to talk about today. So the question is, does the vaginal microbiome impact preterm birth? The first obvious observation that we made was that if we looked at women who were going to deliver preterm, we find that they have a higher, this is reversed actually. That's 5 minutes. Okay, so the women who were going to undergo preterm birth had a lower percentage of vaginotypes that were dominated by Lactobacillus crispus in particular. And this was an observation. We've made this observation. Several other groups have also made this observation that Lactobacillus crispus seems to be associated with a positive outcome or a term birth. Then we asked the question, are there specific taxa that convey risk of preterm birth? And the answer is yes. I mean, obviously I already talked about one, Lactobacillus crispus, but there's a number of bacteria have a negative impact on association with a negative association with preterm birth, and they're listed here. They include things like BVA-B1, Schleithia amnii, and others. One surprising observation that we made was that we didn't see a relationship between Gardnerella and preterm birth. Gardnerella is probably the most highly associated with bacterial vaginosis, and we kind of expected that would be one of the bad players. In our hands, it wasn't, and there are probably reasons for that, which we could talk about if you're interested. We did find that there were 4 taxa that we could use to model preterm birth or risk of preterm birth, and this was a project that Dr. Tom Aradz in our College of Engineering used machine learning to try to predict preterm birth using those 4 taxa that I showed in the previous slide, looking at them early in pregnancy. And from this, he could model and predict preterm birth with around 77% specificity and sensitivity level. So it's not perfect. It's maybe just an advance, but it still says that the microbiome potentially can help to predict outcomes of preterm birth. One interesting observation was this signal was only present in the first trimester, early pregnancy. The signal disappeared, and that probably is because, as I mentioned before, the vaginal microbiome tends to homogenize towards a more lactobacillus or healthy microbiome during pregnancy. So that the signal early in pregnancy is washed out later in pregnancy, but it is obvious that you can see it early in pregnancy. The idea there, I guess, is that in the long run, one may be able to predict preterm birth risk prior to pregnancy. We still haven't been able to do this study where we look at women before they get pregnant and see if their microbiome is similar to their microbiome in early pregnancy, but we assume that early microbiome, vaginal microbiome, probably is closely represented by what is pre-pregnancy, and we're thinking that that could potentially be a way to intervene prior to women getting pregnant. I'm just going to switch for a quick minute. This is Dr. Duda made a very nice presentation of breast milk. We looked a little bit at the contribution of the female, the mother's microbiome to the child, and we didn't really find a whole lot of connection. In this particular slide, we're comparing the neonatal microbiome from day 0 to day 2, so a very short period of time. We know that the neonatal microbiome is dominated by the mom's vaginal microbiome right after birth, within 5 minutes. It's obvious why if it's a normal delivery. But what happens after that? And so we looked at the baby buccal, rectal, and stool samples, compared that to mom's, and what we could see is that the babies kind of the neonates kind of clustered. They tend to cluster distinctly from the mom's microbiomes. There's some overlapping and some overlap of the neobuccal with the mom's buccal. You can see that here. The red is baby buccal, and you can see it interspersed here. You can also see some of the baby stool interspersed with the mom's vaginal. So there is a contribution there. Our point is that what happens after day 0, day 1, that was a day 0 analysis. If you look at day 0 to day 2, here I'm showing a little movie where if you watch the mom's buccal, the mom's samples stay stable and the baby's rectal are moving. And what you see is the black is pretty much diverse, distributed among all the other mom's vaginal, buccal, and rectal microbiomes. So it's kind of not clear exactly what is the major contribution. But if you watch what happens, within 2 days, you start to, the baby rectal microbiome tends to congeal or cluster together. And that's happening within 2 days. And the same thing is true of the buccal, is it starts to be distributed with contributions from multiple different parts of the mom's anatomy. But if you watch it, it congeals into a particular cluster of its own. So that what we're taking this as evidence that there is a very rapid niche specialization from the neonate. It's not, it doesn't take long before the neonate starts to select out what bacteria it's going to have in its particular organs. Okay, I'll stop there. I just want to thank all this 6,000 or 8,000 women who contributed to our study, all the people that participated in it, and obviously all the funding agencies, mostly from NIH, but also from a little bit from Gates and some other organizations. So thank you. Stop there.
Thank you very much, Professor Buck. Please take a seat over here. I think the questions will be taken at the end of the session. So I will request my co-chair to introduce the next.
Thank you, Dr. Reddy. I have the pleasure of introducing Dr. Jeffrey Klossner from the University of Southern California, USA. He's going to be joining us online. And he is the Deputy Health Director and the Director of STD Prevention and Control Services at the San Francisco Department of Public Health, and is a member of the UCSF School of Medicine faculty in the Division of AIDS and Infectious Diseases. He helped identify key factors associated with the increased spread of HIV and STDs and implemented multiple novel public health prevention programs. His research interests are in applied epidemiology and the prevention and control of infectious diseases of public health importance, like HIV, STDs, TB, COVID-19, and cryptococcal infections. Dr. Klossner has received his MD from the Cornell University Medical College and completed his training in internal medicine at NYU Bellevue Hospital. Welcome, Dr. Klossner.
All right, thank you very much. I'm very happy to be joining you all today. I'm sorry that family situation prevented me from being there in person and meeting many colleagues and friends that I have had working actually in India, in South India, and in different areas for many years. So I'm going to share my screen, and hopefully someone's going to let me know if this is correctly working.
So your screen is visible, but if you could just change the mode to the presentation mode. Right now it's in.
All right, yeah, so I need to change the display settings. Which
Yes.
is tricky. Okay. Is that good?
Yes.
All right, great. So today I'm going to give a quick review on sexually transmitted infections, the microbiome, and pregnancy. Greetings from University of Southern California in Los Angeles. Over the past 2 years, have received different consulting fees and grants from a variety of different commercial entities. Wanted to cover some of the epidemiology of early infant mortality, preterm birth, some of current ideas of pathogenesis, and then as a clinical translational scientist, I'm particularly interested in interventions. So what can we do to, given we understand that there are either infections or changes in the vaginal ecology to have an impact on birth outcomes, and then provide a brief summary. So we know that the infant mortality rate varies substantially around the world, and particularly in low and middle income countries, we have a much higher death rate per 1,000 live births. However, many people don't realize that the #1 cause of infant mortality, which is mortality less than 1 year of life, is actually driven by complications of preterm birth. So from a public health perspective, if we can have a better prevention strategy to reduce the frequency of preterm birth, we can have a substantial impact on infant mortality. We know that preterm birth also can be devastating to the mother, to the immediate family. High infant mortality also contributes to higher number of pregnancies, also impacts a woman's ability and the family's ability to contribute economically in the overall population. So a reduction in preterm birth can have multiple outcomes and improve society as a whole. Also know that survival of a preterm birth infant is directly related to the gestational age. We typically think about 3 or 4 different categories of preterm birth, from extremely preterm, very preterm, and moderate to late preterm. A preterm infant born in a high-income setting, such as some parts of the United States, can have mortality as low as 10%, where in some other settings around the globe, preterm birth mortality may be as high as 90%. Preterm birth is a complex issue. We tend to divide it into what's called spontaneous and indicated or induced. So physicians may induce preterm birth for a variety of maternal conditions. The spontaneous preterm birth may be associated with preterm labor or premature rupture of membranes. And then there's a variety of different phenotypes of spontaneous preterm birth, and each phenotype may have subtle differences in the pathogenesis. And that's why when we study the cause and pathogenesis of preterm birth, we're at risk for making errors if we tend to lump all preterm birth as a single entity. There are a variety of different causes of preterm birth, which may be vascular associated, hormonal associated. The ones I focus on in my research are infection related, and it's estimated that infection may cause about half of spontaneous preterm labor, which may lead to preterm birth. But so by focusing on infection, in particular cervical vaginal infections, that's probably where we can get the most impact in terms of preterm birth prevention. And then furthermore, when we look at infection, we think about essentially two different causes of intrauterine infection. There's there is a disseminated infection or bloodborne infection, which may be related to hematogenous spread of things like human immunodeficiency virus, cytomegalovirus, syphilis. And then there's ascending infection, which may be due to bacterial STDs like chlamydia, gonorrhea, potentially even trichomoniasis. Mycoplasma, ureaplasmas have been associated with ascending infection. And then we just heard an excellent talk about dysbiotic vaginal microbiota, or historically have been called bacterial vaginosis, and the role of different organisms, bacterial vaginosis, associobacteria, that may also cause ascending infection. In my work over the past about 12 years, we've looked at the frequency of sexually transmitted infections in pregnancy and their association with ascending infection. And we find globally that there is substantial epidemiologic diversity. So you have highest rates overall in places like Haiti and South Africa, and much lower rates in places like Pakistan, Lebanon, or Bangladesh. And even within these countries, you can see variation by specific organisms. So it was interesting in South Africa, and we continue to work in South Africa, about anywhere between 1 in 4 and 1 in 5 pregnant women will have a chlamydia infection, and about 1 in 20 will have infection with gonorrhea. So these bacterial STIs are actually very common in certain settings. And the mechanism after ascending infection is, has been shown by numerous investigators, and I think we'll hear some further evidence today, is related to the infection causing inflammation. And researchers tend to categorize infection as microbial induced, or from infection when there's evidence of pathogens present, or non-microbial or induced inflammation, which may be due to environmental stressors, even micronutrient deficiencies, and other factors that would be non-microbial related. In our work in Botswana, we've been able to show that there are complex relationships between the presence of chlamydia, the increase or decrease of certain plasma cytokine profiles. So it's fascinating to me that you don't just see the abnormal inflammatory markers in the vaginal mucosa and in the vaginal compartment, which is where we often look, but you can actually see systemic inflammation if you look in the plasma, and these can be mediated by the gestational age and also by HIV infection status. So biomarkers. So we're always looking for different biomarkers that will risk stratify women who may be at risk for preterm birth, and also help us understand mechanisms in terms of pathogenesis. So the primary biomarkers tend to be Th1 associated, such as interferon gamma, IL-2, and IL-12, versus more Th2 cytokines, such as IL-4 and IL-10. IL-6 has been studied more extensively, has a high sensitivity and specificity for intrauterine infection. And then IL-8 has been demonstrated as well, increased in cases of chorioamnionitis. So taking a little bit, deeper dive into signaling pathways. There's a variety of different models that attempt to show that, there may be downstream effects of activation of NF-kappa beta that then leads to these pro-inflammatory cytokines, as you showed, that then leads to infiltration and expansion of neutrophils and associated increase in prostaglandins and contractile proteins in the various tissues. And then ultimately, that leads to cervical remodeling. And it's this really cervical remodeling, these physical changes that may ultimately lead to preterm. So kind of a general model of how infection leads to inflammation. Inflammation may lead to a disruption in the normal fetal-maternal tolerance associated with immune activation and rejection of the fetus, which leads to preterm birth. So these kind of models allow us to think about different points where we may intervene to reduce the frequency of preterm birth or identify markers that may predict preterm birth. So we actually just heard a very nice presentation about the vaginal ecology and the different taxa that have been associated with bacterial vaginosis or dysbiosis and what those associated bacteria may be. And I just pulled one study that was recently published, which looked at a specific subset of organisms and why these organisms may be associated with disruption of this cervical vaginal integrity through the production of proteases. And this is, still early work, but this, suggests that, the certain increase in these BV-associated bacteria through their increased presence and increased production of different enzymes may reduce the integrity of the cervix and the cervical amniotic barrier there. So I just want to move for a few minutes to different interventions, as this is kind of where I focus my work internationally is, okay, well, we have an understanding or increased understanding of some of the mechanisms, but what can we actually do about it? So these are two large trials, the WANTAME trial in Papua New Guinea, which is led by the Kirby Institute out of Australia, funded by the Australian government, and they enrolled 4,600 pregnant women in a cluster randomized trial and offered screening for chlamydia, gonorrhea, and trichomonas and bacterial vaginosis in different strategies to different women in different clinics, and observing these women for an impact on the frequency of preterm delivery or low birth weight. And then the study I'm leading in South Africa is a study of 2,500 pregnant women in Eastern Cape, where I showed you a very high prevalence of bacterial STIs and Trichomonas vaginalis. About 40% of pregnant women have at least one of those three curable organisms, chlamydia, gonorrhea, or Trichomonas. And in this trial, it's a 3-arm trial where we're comparing to the standard of care, screening and treatment, a single event, screening and treatment twice in pregnancy, and also a test or cure versus standard of care. So both these studies should be reported out shortly. The WANTAME trial probably in the next couple months, and then the South African trial within the next year. And then lastly, because as I mentioned, preterm birth is actually multifactorial, it's quite complicated. This is the newest large intervention trial for preterm birth prevention called the AAA trial. And this is a combination intervention. It will use what we call a poly pill So we'll be combining both aspirin and vitamins or micronutrient supplementation into one pill. And the aspirin and the micronutrients will be given every day, and the azithromycin once at enrollment. And this study is funded by Open Philanthropy, which is a relatively new global health philanthropy that's based in the United States. And the rationale for this combination intervention is that there's good evidence that anti-inflammatory low-dose aspirin reduces preterm birth, that in this study of over 12,000 pregnant women called the aspirin trial in 2020, they were 11% less likely to develop 37 weeks in the low-dose aspirin arm and 25% less likely to deliver 34 weeks. The suggested mechanisms are through the aspirin effects of decreased inflammation with or without changes in thrombotic mediated pathways. The second component is the antenatal micronutrient supplementation, and that was studied in a very large cohort of over 96,000 pregnant women in Botswana, which actually will be the study site of this new AAA trial, where they looked at pregnant women who received more comprehensive micronutrient supplementation versus only iron and folic acid. And in this study, they saw a 37% decrease in the frequency of preterm births between the group receiving the micronutrients versus those who only received iron and folate alone. So our study team thought, okay, well, you get a good signal with aspirin, you get a good signal with multivitamins, why not combine them into a single poly pill? And then because we are an infectious disease group, and there's been a lot of work done on different antimicrobial approaches, including work in Egypt that showed that monthly azithromycin resulted in significant higher gestational age and significant higher weight at delivery. Different systematic reviews in malaria endemic settings showing that azithromycin was effective at reducing preterm and increasing birth weight. And then some early work in Papua New Guinea, which actually also showed that azithromycin in combination with anti-malaria medication resulted in lower inflammatory markers and a substantial reduction in preterm frequency. So this AAA polypill study design will be a randomized 4-arm placebo-controlled trial and have additional components such as a cost evaluation, process evaluation, and specimen collection for mechanistic studies, which means we'll be collecting blood and vaginal swabs to look at changes associated in the different intervention arms. So in summary, infant mortality remains high globally. There are several preventable or treatable conditions which still remain poorly addressed. Preterm birth is a major problem. It's multifactorial. The pathogenesis of preterm birth is beginning to be understood, and various intervention studies are underway. I want to thank you for listening today. And again, my apologies for not being able to make it in person. Thank you very much.
Thank you, Dr. Klossner. We'd request you to stay online as we'll be having the questions at the end of the session. Thank you.
Good afternoon. It is indeed a pleasure and honor for me to invite Dr. Douglas Cohn to deliver his lecture talk. Dr. Cohn is a physician scientist and director of clinical operations at the Ragon Institute, MIT, and Harvard. He is also doing clinical practice in the Division of Infectious Diseases at Massachusetts General Hospital. He received his MD and PhD degrees from New York University and underwent internal medicine training at the University of California, San Francisco, and New York Hospital, Weill Cornell Medical Center. Dr. Cohn completed his training in combined Massachusetts General Hospital and Brigham and Women's Hospital Infectious Disease Fellowship Program. His research focuses on understanding the role of mucosal tissues in HIV accusation and disease progression. His lab is involved in developing and applying new technologies to overcome the technical barriers that have impeded our understanding of mucosal immunity. Welcome, Dr. Cohn. It's all for you.
Great. Thank you for that very generous introduction. And it's a real pleasure to be here today to speak to you. It's been really exciting to talk to many new investigators here and also hopefully future collaborators. So I'm going to be talking a little bit about our work on the vaginal microbiome and its impact on specifically genital inflammation and how that then influences reproductive health outcomes. And we first came about our interest in the vaginal microbiome through an interest in HIV infection. And it was very clear that globally, if you looked at new HIV infections, most of these were occurring within sub-Saharan Africa. Most of them were occurring in young women of reproductive age. And it's very clear that there's an unequal burden of the HIV pandemic on young women in sub-Saharan Africa, where if you look at HIV prevalence rates, it's up to 8-fold times higher in young women than young men in many of these countries in sub-Saharan Africa. And so that really drove us to try to understand what were the factors the biological factors that were impacting the risk of young women for acquiring HIV. So we started a study called the FRESH study, and FRESH stands through Females Rising Through Education, Support, and Health. This study has enrolled over 3,600 HIV uninfected young women aged 18-23 in the township of Umlazi, South Africa, which is just outside of the city of Durban. The women come to the site twice a week. They get a finger prick viral load test each visit, so we can determine when women acquire HIV with very high precision. And they also have routine pelvic exams every 3 months with larger blood draws. And they don't come to the site just to participate in research, but they also engage in a poverty alleviation program where they spend 3 hours each visit there in the clinic learning job skills, learning how to use computers, learning how to create a resume, in addition to obviously HIV prevention and health maintenance, an HIV prevention curriculum. And we're very proud of the fact that after graduation, being followed for 10 months, that the majority of these participants actually go on to meaningful activities like either jobs or internships or completing their schooling. And yet the program, despite the intensive educational program about HIV prevention, does not reduce the HIV acquisition rates beyond those in the community. And to just get a better understanding of what that looks like, within sub-Saharan Africa, South Africa, Southern Africa in particular, has very high HIV acquisition rates. And within Southern Africa, South Africa, and particularly this region of KwaZulu-Natal, which is where our study base, has enormously high HIV incidence and prevalence. So in the Vukuzazi study, which did a door-to-door community surveillance of over 20,000 individuals living in a community of 40,000, where they did HIV testing of all the participants, they saw that amongst 14-year-old girls, less than 1% were HIV infected. And by the time women were in their late 20s or early 30s, over 60% had acquired HIV. So you can see that in some regions of the world, HIV continues to be unabated, with HIV prevalence rates that are really astonishing, particularly amongst young women of reproductive age. So, but there are reasons to be optimistic about efforts to prevent HIV transmission in young women. And part of that is because of the biology. HIV transmission is actually relatively inefficient. We know that only about 1 in 500 exposures result in an HIV acquisition event. And when women are exposed to HIV, they're exposed to enormously diverse quasi-species of viruses. And in most cases, it's a single virion that's able to break through these natural barriers within the mucosa of the female genital tract to infect a local activated CD4 T cell there within the tissues of the female genital tract. And then there's this period of local obligate expansion there within the tissue until you have sufficient burden to then disseminate. And once you've disseminated, you've kind of created this long-lasting latent viral reservoir that becomes very, very difficult to eradicate. But that initial period of local expansion within the tissues of the female genital tract is thought to take between 7 to 10 days. And that's been referred to as window of opportunity, because if we can stop HIV replication there within the tissues, we can prevent systemic dissemination and prevent establishment of infection. So our lab has been really interested in understanding what are the biological factors that affect the efficiency of that initial process there in the female genital tract after women are exposed to HIV. And a really important clue came from post hoc analysis of the CAPRISA-004 trial. Now, the CAPRISA-004 trial was a trial of a intravaginal gel containing the antiretroviral tenofovir. And that product was shown to have a partial effect in protecting women from HIV. So 39% protection. But what was seen in post hoc analysis was that women who had higher levels of genital inflammation, so that was measured by increased levels of inflammatory cytokines within genital secretions, had an over threefold higher risk of acquiring HIV infection. So that really identified genital inflammation as an important risk factor for HIV, but no one really understood at the time what were the drivers of that genital inflammation. So one of the first things that we did in FRESH was to look at inflammatory cytokines within genital secretions. And so in this representation, each column is a different participant, each row is a different inflammatory cytokine, and this heat map is on a log scale. And what we saw was that although all of these women were clinically healthy, women had a very broad range of genital inflammation. And so women in this highest quartile actually had up to 1,000-fold higher levels of some of these inflammatory cytokines than women in the lowest quartile. And so one obvious question at the time was, well, maybe this is driven by known factors that can affect genital inflammation, like sexually transmitted infections, as we heard from Dr. Klausner. But these women with high levels of inflammation, only a quarter of them actually had an STI. So most of these women actually had high levels of genital inflammation for uncertain reasons. And we looked at various behavioral and demographic factors like contraceptive use, sexual behaviors, partners, and there is no clear associating factors that would explain why these women had high levels of genital inflammation. And that's where we started our studies on the vaginal microbiome. So here, as you've seen from Dr. Buck, this representation shows sort of different vaginal bacterial taxa in different colors, and then each column again is a different woman. And what we saw was that generally the community types that we saw within our cohort of South African women fell into sort of four groups, which we called cervicotypes or CTs. So there was CT1, which is sort of the classically optimal Lactobacillus crispatus cervicotype, which actually represented less than 10% of the women. Then about a quarter of the women actually had a cervicotype that was dominated by Lactobacillus inners, which is there in green. But most of the women, over 60% of the women, actually had these anaerobe dominated communities that had very low lactobacillus, which has been referred to as dysbiosis, and also some forms of are actually bacterial vaginosis. So although these women were asymptomatic, many of them had these quote unquote dysbiotic communities. And what we saw was that these very closely associated with genital inflammation. So women with CT3 communities had levels of inflammatory cytokines, looking either in aggregate or at individual cytokines. And in fact, having a CT3/4 type community was a better predictor of genital inflammation, inflammatory causes like chlamydia or gonorrhea. These women with these anaerobe dominated community CT3/4 also had increased numbers of activated CD4 T cells there in the cervix. And these again are thought to be the first cells that are infected with HIV. And so we predicted that these women with CT3/4 type communities would actually acquire HIV at higher rates. And when we looked prospectively, that's what we saw, that women with CT3/4 type communities, these anaerobe dominated communities with low lactobacillus, were acquiring HIV at over 4-fold higher rates than women with Lactobacillus crispatus. And that was even accounting for sexual risk behaviors or sexually transmitted infections. And as has been talked about very nicely by both Dr. Buck and Dr. Klausner, we were really interested in sort of like, well, what are the host cells that are sensing these inflammatory bacteria, and what are the specific cellular pathways that are being activated? And so when we looked at a number of different lineages through transcriptional analysis of cervical cells, what we saw was that antigen-presenting cells had the most differentially expressed genes in women with Lactobacillus crispatus versus CT3/4 type communities. And LPS signaling was one of several signals that we saw upregulated in these antigen-presenting cells. And what was interesting was that these many of these same signaling pathways that we saw associated with these anaerobe dominant communities had also previously been described in the preterm birth literature as also being associated with preterm birth. Things like IL-1 beta, TNF alpha, LPS, NF-kappa beta, NF-kappa B, and IL-6, IL-8 were all associated with preterm birth, and all were also upregulated within our studies. And so it seems that there's sort of these candidate pathways which sort of show are upregulated with these diverse anaerobe dominated bacteria, as well as preterm birth, and has been noted by a number of speakers today. These same diverse anaerobe communities are actually associated with a number of poor reproductive outcomes, which largely have a greater impact in low and middle income countries. So we also furthered this because we had an opportunity to follow these women for over 10 months. We could look longitudinally at what was going on. And one of the things that we saw was that when women transitioned from a Lactobacillus crispatus dominant community to one of these anaerobe dominated communities, they generally went through a transition state of Lactobacillus iners dominance. And so Lactobacillus inner dominant states seem to be this promiscuous state that facilitates the transition from more optimal Lactobacillus crispatus to these anaerobe dominated communities. And that kind of made sense with something that had been known in kind of the clinical realm for some time, which was that when you treat women with bacterial vaginosis with the standard of care antibiotic, metronidazole, what you do is you reduce all of those anaerobes, but then you greatly increase the abundance of Lactobacillus iners. And we saw that also within our studies, where in blue, when you have women with BV, and then you treat them with metronidazole, you shift them all to green Lactobacillus iners, and then over time, there's a very high probability that they then revert back to these BV-like communities. So what was really needed was an intervention that could sort of break out of this cycle of going from iners to BV and back to iners, and ideally would be able to kind of shift iners dominant communities to Lactobacillus crispatus dominant. And so that was something that we thought would be really useful as potentially an adjunctive therapy, keeping in mind that standard of care treatment for BV has not been for over 4 years. So there is clearly a real need for innovation in this area. So we looked at many different factors, but one sort of factor that we're really interested in were unsaturated long-chain fatty acids, in part because these compounds are very much enriched at mucosal surfaces. Most bacterial systems have specialized ways in which they sense, acquire, and utilize exogenous fatty acids. But there are also interesting examples where unsaturated long-chain fatty acids can also specifically inhibit the growth of specific bacteria like palmitoleic acid and Staph aureus. And what we found was that, in particular, oleic acid and structurally related unsaturated long-chain fatty acids can actually inhibit the growth of Lactobacillus iners and promote the growth of Lactobacillus crispatus. And this inhibition of iners seemed to be bactericidal when we looked at sort of the ultrastructures of Lactobacillus iners treated with oleic acid and other unsaturated fatty acids. So we next performed bulk RNA sequencing on cultured bacteria that had been treated with either unsaturated long-chain fatty acids or untreated, and looked for shared differentially expressed genes. And we've only found 3 unique genes that were commonly upregulated in cells in all these different types of Lactobacillus, crispatus, gasseri, and Jensenii, when they were treated with multiple different unsaturated long-chain fatty acids. And those were an oleate hydratase, and then a putative fatty acid efflux pump called FAR-E, and then a gene that was reported to regulate FAR-E. And using a collection of over 1,000 different female genital tract lactobacilli that we'd sort of accumulated and also sort of scoured from publicly available sources, both genomes and mags, we found that oleate hydratase and FAR-E specifically were uniformly absent in all strains of Lactobacillus iners that we identified, that we looked at, but were actually present within other non-iners lactobacilli. And so we thought that, this fatty acid utilization through one of these genes could actually explain why there was this differential effect of oleic acid in particular on Lactobacillus inners and other non-Lactobacillus inners bacteria. And so what we did was then we knocked out these two genes in Lactobacillus gasseri. And we showed that, in fact, that if you knocked out FAR-E, this putative transporter, that you could actually inhibit the growth of Lactobacillus gasseri, or make it more sensitive to bactericidal effects of oleic acid. Additionally, using labeled oleic acid, we showed that these non-inners lactobacilli are able to use oleic acid and incorporate into sort of structural lipids within the cells, and not necessarily use them as metabolic substrates for producing ATP. So that brought us back to sort of the original clinical question, is like, could this be used to then actually help treat things like bacterial vaginosis and facilitate the creation of Lactobacillus crispatus dominant vaginal communities? And so we created synthetic BV-like communities by mixing various primary isolates that we collected from women in the FRESH program. And then we combined these, and we showed that, yes, if you treated with metronidazole, you could see the predicted increase in Lactobacillus inners. But if you treated with oleic acid, either alone or with metronidazole, you could promote these Lactobacillus crispatus dominant communities. So we think that this could actually be one way to perhaps augment standard of care treatment with metronidazole. So just to kind of summarize the talk, when you have Lactobacillus crispatus, you sort of have a low inflammatory state in the genital tract. But when these communities are displaced by these anaerobe dominated communities with low Lactobacillus abundance, you have high levels of inflammatory signaling by local antigen presenting cells. You have proliferation and recruitment of activated CD4 T cells to the cervix and vagina. And that when women are exposed to HIV, that increases their biological susceptibility to establishment infection. But in addition, there are also a multitude of other different effects of these anaerobe dominated communities, including effects on mucin and mucus, IgA cleavage, the production of immunomodulatory molecules, and others. So with that, I'll just stop and thank the people who've done the work, and thank you for your attention.
Thank you, Dr. Korn, for this wonderful presentation. I request you to come and sit over here. Oh, there's a seat over there.
I request Dr. Vrati to introduce the last.
Thank you. The last speaker is Daisy Talukdar. Dr. Talukdar is a research scientist at THSTI. Her current research is focused on microbial genomics and microbiome study in the ongoing project, that is Interdisciplinary Group for Advanced Research on Birth Outcomes, DBT India Initiative, that is Garbhini, phase 2. And to study the functional insights of different lactobacillus species isolated from the vaginal milieu. She has an expertise in genomics and metagenomics, data analysis, and published her work in reputed journals like Genomics, Gene Frontiers, etc., etc. She has also worked in various other projects where she analyzed the role of microbiome in COVID-19 severity, ventilator-associated pneumonia, and lung and gallbladder cancer. Dr. Talukdar, please.
Thank you, sir, for the introduction. Good afternoon, everyone. So to begin with, the human gastrointestinal tract, the major site for micronutrient production, is populated with trillions of microbial cells from all the 3 domains of life. Several factors like diet, antibiotics, host genetics can modulate the gut microbiome, thereby leading to dysbiosis, further inflammation, and several health disorders like malnutrition, diabetes, colorectal cancer, so on and so forth. The diversity and the composition of the gut microbiome of healthy Indians was not well understood. So in 2018, we carried out a study where we wanted to study the gut microbiome of healthy Indians in 3 different communities. So from our study, we found Firmicutes to be the most abundant phylum, followed by bacteroids, proteobacteria, actinobacteria, Verrucomicrobiota, and the last is the fusobacteria. So coming to today's topic of discussion, the preterm birth and its burden in India. As we all know that preterm birth is one of the major causes of infant mortality as per the UNICEF. India contributes to about highest, that is 3.2 million per year, as any of the developing countries of the world. And it has been hypothesized that the vaginal microbiome is associated with preterm birth. We have few evidences before carrying out the study, like the vaginal microbiome is one of the least diverse microbiome in the human body. It has been grouped into 4 community subtypes. CST1, which is associated with term birth, that is CST1 belongs to Lactobacillus crispatus, and CST4, which belongs to non-lactobacillus species, is found to be associated with preterm birth deliveries. However, we do not have a clear picture on the relationship of the vaginal microbiome composition and its association with the birth outcome. So our study is a part of the large pregnant women cohort Garbhini, a DBT India initiative. Till now, 7990 women have been enrolled in Garbhini. So there were few inclusions and exclusion criteria when we were enrolling these women. Inclusion criteria like women whose gestational age was less than 20 weeks were enrolled in the study, and women who agreed to deliver in Gurugram Civil Hospital were also included in the study. And exclusion criteria like women taking antibiotics prior to 7 days before the sampling were excluded, and women having complications during the pregnancy like preeclampsia, placental abnormalities were all excluded from the study. So collection of HBS sample, we collected in the first trimester, that is less than 14 weeks, second trimester 18 to 20 weeks, and then third 26 to 28 weeks, which was followed with metagenomic DNA isolation, followed by sequencing of V3, V4 region of 16S rRNA gene in HiSeq 2500. Then we did a pilot study. That is, previously we did a pilot study where we included 18 preterm samples and 20 term birth samples, followed by validation of the pilot study where 60 preterm samples were included and 140 term birth samples were included. This was followed with development of a rapid dipstick assay. So these are the observations from our pilot study, which we conducted previously. So here we can see that the alpha diversity indices are almost similar in both the term and the preterm. However, Lactobacillus iners was found to be significantly high in the preterm samples, and it was less in the term birth samples, whereas Lactobacillus crispatus and Lactobacillus gasseri was found to be in high abundant in the term birth samples, and it was less in the preterm samples. We also did an analysis of the community state subtypes, where we identified CST1, as I already mentioned, which belongs to Lactobacillus crispatus, was predominantly high in the term birth samples, whereas CST3 and non-lactobacillus species was predominantly high in the preterm birth samples. Additionally, we also observed CST2 to be only present in the term birth samples, and it was totally absent in the preterm samples. Other bacteria like non-lactobacillus species like Gardnerella, Megasphaera, Snithia were also very high in the preterm birth samples, and its abundance was less in the term birth samples. So we published this work in 2021 in Frontiers in Cellular Infection Microbiology. Next, we proceeded to validate the findings of our pilot study by increasing the sample size to 600. So here we observed that the alpha diversity was significantly altered in the first trimester between term and preterm. It was high in the preterm samples and it was less in the term samples. And the alpha diversity also significantly varied within the preterm in all the 3 trimesters. We then did a core species analysis, taking 75% as the threshold criteria and relative abundance of 0.1, less than or equal to 0.1. So here we identified Firmicutes to be the highest abundant phylum, followed by Actinobacteria, Proteobacteria, Bacteroideata, and Fusobacteria. In the genus level, Lactobacillus was seen to be predominant in both term and preterm samples. Next, we studied the vaginal microbiome composition in early pregnancy between term and preterm delivering mothers. Here we observed Lactobacillus iners to be significantly high in the first trimester in the preterm birth samples, whereas Lactobacillus crispatus was significantly high in the term birth samples in the first trimester of pregnancy. This was followed by a Lefse analysis where Lactobacillus crispatus and Lactobacillus johnsoni was predominantly high, or we can see it was significant in the term birth samples, whereas non-Lactobacillus species like Snithia, Prevotella, Epicobium, Pseudomonas, and Lactobacillus iners were predominantly high in the term birth samples. We also did a community state subtype analysis, and our results correlates with the results of a pilot study. So similarly, like the pilot study, here also we observed that CST1, that is Lactobacillus crispatus, was predominantly high in the term birth samples, and Lactobacillus iners and CST4, that is non-Lactobacillus species, were predominantly high in the preterm birth samples. Next, we also wanted to study the association, or we wanted to see the relationship between different microbial species. So our correlation analysis reveals Lactobacillus crispatus to be negatively correlated with Lactobacillus iners. We also studied the correlation between Shannon diversity index and other microbial species. So here we found that Shannon index was positively correlated with non-Lactobacillus species, and it was negatively correlated with Lactobacillus iners. So till now, the summary of our study are L. crispatus, L. gasseri, L. jenseni are high in the first trimester of the term birth samples, whereas Lactobacillus iners, Gardnerella, Megasphaera, and Snithia are high in the first trimester of the preterm birth samples. Now, last year, or we can say earlier this year, we developed a rapid point of care dipstick assay to identify and differentiate SARS-CoV-2 variants in COVID-19 positive patients. So we applied the same principle in our preterm birth population to further develop a rapid detection of preterm and term birth associated bacterial taxa using dipstick-based chromatography. So in the first image, as you can see, Gardnerella vaginalis, Megasphaera type 1, Lactobacillus iners, and Snithia, the predominant taxa which were identified in both the studies in the preterm samples could be seen as a blue test line in the dipstick strip. And in the second image, you can see Lactobacillus gasseri, Lactobacillus jenseni, Lactobacillus iners, and Lactobacillus crispatus, which were identified again in both the studies as the signature bacteria of the term birth delivering mothers, could be seen as a blue test line in the dipstick strip. Now, the beauty of our dipstick strip is that it has a specificity and sensitivity of almost around 70%, and which is much higher than the other biophysical methods which are regularly used, like uterine examination or measurement of the cervical length through endovaginal test and all, etc. So lastly, to summarize my today's talk, the microbial signatures in HBS samples showed Lactobacillus genera to be highly abundant in the term birth samples. The abundance of Gardnerella, Ectopodium, Snithia are found to be high in the preterm birth samples. L. crispatus was the species significantly higher in term birth compared to preterm birth, whereas L. iners was found abundant in preterm birth cohort. The developed potential point of care dipstick assay has a specificity and sensitivity of 67 and 74% respectively. The current findings can help in reducing preterm birth by modulating composition of the vaginal microbiome, either by microbiome transplant or delivering preterm birth associated lactobacillus taxa in the vaginal milieu. So lastly, I would like to thank Executive Director, THSTI, Professor Shinjini, Dr. Bhabtosh Das, Professor Nitya, Dr. Shobik Mukherjee, Garbhini Study Group, NIBMG Kolkata, Funding Agency, Department of Biotechnology, and to all members of Functional Genomics Laboratory. Thank you so much.
Thank you, Daisy. So the floor is now open for questions. We don't have much time, but maybe 10 minutes or so. Yes, Prasanji.
So I have three questions for bits. So can I tell the questions? So then, for Jeff, my question is that actually this is very hard at the end. We forget what the speakers told. So this is the problem. So correlation between that microbiome profiles and the immune data. So that's actually I was missing that. Is there any correlation between microbiome data and the immune data, what Jeff had shown, and also that besides anti-inflammatory therapy, that you were suggesting anti-thrombogenic therapy. So did you find any thrombogenic profiles? Did you profiles any thrombogenic factors in the vaginal samples?
Okay, so let me clarify. So the study about the
So this is for Jeff. So if it's
there, let me just check.
Okay.
Can people hear my
Can
response?
someone just check if Dr. Klossner is there?
Yeah. So another question to Greg is that so this is a very nice study that preterm vaginal microbiome types, which actually you found that lactobacillus, which is so. Is there any correlation? Is there any correlation that so? Actually, I'm missing that. Is there any correlation with the inflammation and all these things? Did you find?
Correlation with the production of what?
Inflammatory factors.
Yes, sir. We didn't report on that, but in our analysis, we looked at a number of cytokines. I can't recite them from memory, but there were a number of cytokines that were associated with risk of premature birth. Yes.
Okay. So, Jeff, this is a question for you. So that nice study, and actually, so first question is that it's a very good study. At the end, you were suggesting that anti-inflammatory, anti-thrombotic, alongside nutrients supplement for the preterm patients. So. Did you? I was missing that. Did you profile any thrombogenic factors in the vaginal sample?
So the answer is no. You did not miss it because the study has not started yet.
Okay. So, but this is a very nice combination. You found that therapy, anti-inflammatory, along with anti-thrombotic, which is aspirin you are using. And so this is, then how did you come up with this solution, that anti-thrombotic?
Right. So that's from other studies of other scientists who have studied aspirin and found a substantial benefit. And in those studies, they also did not measure any specific thrombotic effects. So you're going to have to invite me back in 4 years. And in 4 years, I will present to you the thrombotic measures and convince you how the aspirin is reducing preterm birth.
I wish that after 40 years, I would like to see you.
4 years, 4 years.
Oh, 4 years. Okay. I'm sorry. Yeah, sure. I look forward. So another question for Dr. No. Very nice study, and I'm really just would like to appreciate that. So do you think that the compound what you are using can be useful for the therapy for preterm birth patients as well?
Possibly.
What you are using?
Yeah, I do think that there are clear associations between
Yes.
vaginal microbiota composition and a number of adverse outcomes, including preterm birth. And so if we could durably alter communities so that they were Lactobacillus crispatus dominant, the prediction would be that there would be an improvement in preterm birth outcomes as well as other reproductive outcomes, including sort of HIV. So that would be the hope, but that remains to be seen.
Yeah, excellent. Thank you.
My question is to Dr. Buck. Like you showed that lactobacillus increases during pregnancy, right? So have you studied like post-pregnancy, whether it comes to the normal or it remains like that only?
Can you rephrase the question?
Actually, lactobacillus species that increases during the pregnancy, that is your finding. So post-pregnancy, does it reverse to the normal or it remains like that only?
That's an interesting question. Post-pregnancy, it's been reported by a number of investigators that the microbiome becomes chaotic, more or less, much less lactobacillus dominant, so much more complex than normal. So, and it takes a while for that to return, several months, if not year, for that to come back. So, I mean,
I have actually a related question to you. You showed that there was higher abundance of lactobacillus in pregnant women as opposed to not pregnant. Now, is it the higher preponderance is cause or effect of pregnancy?
Yeah, I think it's because of the hormones that are being expressed during pregnancy is changing the
after that.
Right. So I actually, yeah, that's probably the most likely explanation.
I was just wondering whether women who have higher preponderance of lactobacillus, they are more likely to get pregnant.
That is a good question. I mean, people are looking at infertility in terms of the microbiome, and there is some evidence that suggests a more healthy vaginal microbiome may be contributing to the ability to get pregnant, right?
It's actually, there is some, as Greg was saying, there is some data suggesting that with in vitro fertilization, you have higher rates of successful embryo transfer and implantation when women have Lactobacillus crispatus. So, yeah, Lactobacillus crispatus.
Hello, I have a question for Dr. Desi actually. So as you mentioned that here.
Yeah.
So as you mentioned that vaginal microbiome transplant can help for the preterm to the term birth. So what is your thought regarding just having a single capsule, having single bacteria of Lactobacillus crispatus as it is playing the major role?
Thank you. Wonderful question. Actually, we are working on this. So this is a preliminary study. After that, we are working on the in vitro testing, like in the lab. And we are also trying to explore the genomic insights of lactobacillus, which are isolated from the term birth samples. So yeah, we are thinking to develop a consortium, and yeah, hopefully in the next few years, I can reply to your question.
Thank you.
Hello,
Yes,
my
one-
question.
Go ahead, whosoever has the microphone.
Okay,
please.
my question is Dr. Buck. Buck, I think I'm putting the name wrong. Second from the left, I think. Yeah.
Yeah, go ahead.
Yes, my question is, you showed the T-SNP plot between the mother and the newborn child.
No, keep the microphone away from you.
Okay, sir.
Yeah, because it's yeah.
The T-SNP plot between the mother's microbial diversity and the child, early 2 days only. You show na two different clusters. One
We're
cluster
not
for the
clear
mother.
about the question. So,
Are you not getting my question?
Nitya, can you understand the question?
No, I can't. And whom is it addressed to?
No, I think it would be best that you interact with the speaker
okay,
during the lunch, because then you can clearly explain. Yeah, just over the front, just one in the front.
Douglas, so I have just one quick question. So you just reported that increased abundance of lactobacillus is inversely associated with the inflammations. So do these lactobacillus produce any anti-inflammatory molecule, or do they just simply reduce the diversity that cause the reduced inflammations?
You will hear about this afternoon from Dr. Gopinath. Actually, she will be talking about this exact topic.
Yes, please go ahead.
This question is for Dr. Kloster. In your experience, do you see any correlation with MGen and preterm birth, and also asymptomatic bacteria or UTIs?
Yeah, so the answer is yes, between Mycoplasma genitalium. Also, there's been some studies with Mycoplasma hominis as well, and some other uriplasmas. The data is not as strong as with chlamydia and gonorrhea, because there haven't been a large number of studies, and I'm not aware of actually any intervention trials that look at screening treatment for Mycoplasma species to reduce preterm.
Thank you.
Well, I guess in the interest of time and everybody waiting for lunch, so we will close it over here. On behalf of my co-chairs, I would like to thank all the speakers of the session for their very clear, fantastic talks, and the audience for their interaction. Thank you very much. We will close now.
Thank you to all the session chairs for moderating sessions and the speakers for the overwhelming amount of information rendered. Now I request session chairs to kindly felicitate the speakers. I request Dr. Sudhanshu Vrati to hand over the token of appreciation to Dr. Gregory Buck. I request to hand over the memento Dr. Nitya Wadhwa to hand over the memento to Dr. Douglas Swan. Dr. Shibnath Majumdar to hand over the memento to Dr. Daisy Talukdar. Now I humbly request Dr. Shibnath Majumdar to hand over the memento, Dr. T. Ramamurthy to kindly come up on the stage to felicitate the session chairs. Sir, please. To Dr. Sudhanshu Vrati. To Dr. Nitya Wadhwa and Dr. Shibnath Majumdar. I request the session chairs and speakers to assemble for group photo. Thank you, ma'am. Thank you, sir. We now break for lunch. I request the conference volunteers to assist the dignitaries to the area of lunch. We request the participants who are presenting the poster to be present near their respective posters for evaluation. Let us assemble back by 2 p.m. for the start of the next session. Thank you.
कैमरा घुमा नहीं रहा जा रहा देखा होगा ना पीछे एक पॉइंट पकड़ लेते हैं थोड़ा था ना उसमें सेंटर पर पकड़ गया केवल तेरा साइड ही रही है नहीं वो थोड़ा वाइड करी था
Let me check here. No, no, no. Just checking, not changing. That one I think is online. No.
A very good afternoon to everyone. We will be shortly starting our session, so I would request everyone to convene back in the auditorium. Thank you. I humbly request the participants to kindly gather back in the auditorium as we'll be shortly starting our session. Thank you. A very good afternoon to everyone. I would again request the participants to convene back in the auditorium as we'll be shortly starting our session. Thank you. Welcome back, everyone. We now begin with our last scientific session of the two-day conference on the theme, Bioinformatics and Artificial Intelligence for Microbiome Study. I humbly call upon the session chairs, Dr. Shubhra Chakraborty, Dr. Amit Awasthi, and Dr. Ramesh K. Goyal on the stage. Dr. Jenny Kwan will be giving her presentation virtually. Before the talk of Dr. Smita Gopinath, the YouTube streaming will be stopped as requested by the last two speakers. Please. Dr. Shubhra Chakraborty is the director of the National Institute of Plant Genome Research. Her research interests include nutritional genomics, plant immunity and stress genomics, and delayed fruit softening. She has several awards and fellowships to her credit: Inspiring Woman Engineer Scientist Award, Tata Innovation Award from the Department of Biotechnology, Government of India, and the J.C. Bose National Fellowship, SERB DST, are some of them. It is a privilege to have you here, ma'am. Dr. Amit Awasthi is presently working as a senior professor at the Translational Health Science and Technology Institute, Faridabad. He has joined THSTI as faculty After his postdoctoral training from Brigham and Women's Hospital and Harvard Medical School, Boston, Massachusetts. His research focuses on the play of effect on regulatory T cells in IBD and gut infection, understanding T cell differentiation pathways in immunity, autoimmunity, and cancer immunity. He has several honors to his credit, and to mention a few are prestigious awards like the CDRI Award 2022 in the Life Science category for drug discovery, and GN Ramachandran DBT Bioscience Award 2021 for career development. Welcome, sir. Dr. Ramesh K. Goyal is the Vice Chancellor of the first Pharmaceutical Sciences and Research University of India, situated in Delhi. He was previously the Executive Director, Research and Strategies at Viklin Biolabs, Sri Ramachandra University, Chennai. He has over 40 years of experience in teaching and research. He's a recipient of 71 awards, including Best Pharmacy Teacher and Best Pharmaceutical Research Scientist from APTI, and Lifetime Achievement and Distinguished Service Awards from the International Academy of Cardiovascular Sciences, Canada. Welcome, sir. In this session, each talk will be of 15 minutes. After the talk of all the speakers, there will be a 10-minute long question and answer session. I request the audience to kindly reserve their questions till the end of the session. I now request the session chairs to kindly introduce the speakers and call them over for the talks.
A very good afternoon to all. On behalf of my co-chairs, I would like to first thank Thisti for giving this opportunity to chair this session for the wonderful international conference on the antimicrobial resistance and human microbiome. Well, we, as already pointed out, we have 4 speakers, and it has been already told about the time limits. This bioinformatics and artificial intelligence for microbiome study is a real, very interesting and the need of our for the translational research. Especially the importance has come up after the COVID pandemic. How important are these diseases, infectious diseases? And it is not one country or so. Everybody has to come together to see that. So with these small remarks, without wasting any time, I call upon the first speaker, Dr. Tarini Shankar Ghosh from the Triple ITD Indraprasth Institute of Information Technology, Delhi. Dr. Ghosh. Dr. Ghosh is a computational biologist there. He's assistant professor there and PhD from University of Hyderabad. From there, he worked at the Genome Institute of Singapore as a postdoc fellow, and then APC Microbiome at the Cork as a senior postdoctoral scientist. He is an accomplished scientist and recipient of Yakult Young Investigator Award in 2014, followed by Torrent ISHR Award of the Young Investigator in 2017, and was selected for the Pathway Investigator Grant awarded by the Science Foundation Ireland and Irish Research Council. He is also the author of multiple United States or European Union-based patents, and primarily pertaining to these metagenome informatics, algorithms, and identification of putative therapeutic novel microbial consortia targeting fertility and colorectal cancer. Currently, he is working at IIT Delhi, wherein they are trying to search different modalities for the disease. Dr. Tarini Ghosh Shankar, please. He is going to speak on the topic bioinformatics and artificial intelligence for microbiome study.
So good afternoon, everyone. So I hope I don't bore you immediately after the lunch session. So I am Tarini, like sir mentioned, and I'm assistant professor in the Institute of in the Indraprastha Institute of Computational Biology, in the Indraprastha Institute of Information Technology. So I'm basically a computational biologist who works on the human microbiome. So regarding this, so I'll be presenting one of my snapshots of one of the recent research works from our lab, which we are planning to submit soon. And the topic of my talk is in search of the hacks of the adult human gut microbiome. Now, what do you mean by the hacks? Hacks are essentially the health-associated core keystones. So this is a study that was done by one of my MTech students who is just going to graduate in the next few months. And I'll be just presenting some snapshot of the study. However, if you have doubts, we have done some more analysis and we're happy to resolve them as we go. So the human microbiome, as we all know, the human gut microbiome it has many facets, right? It can, as a sensor, it can sense something going wrong in the host physiology and function and can respond accordingly. It can also directly influence the host physiology as a modulator and can be a causative agent of the disease, different kinds of diseases. And at the same time, the human gut microbiome is influenced by a range of different environmental factors like our genetics, our birth mode, our geography, the diet that we eat, the medications that we take, how the microbes that can be transmitted from from the external sources. So the effects of all these on our health, it acts as a translator or a modulator. Okay. Now the thing is, the good part is the human microbiome is therapeutically modifiable, and because it is therapeutically modifiable, that makes it more interesting because then it gives us the possibility of actually therapeutically exploiting the microbiome. And that requires understanding of the hallmarks of what is a healthy microbiome. Now, there are different evidences in this regard onto the patterns of what could the healthy hallmarks of a microbiome could be. Like in this particular perspective by Janvier et al. In Nature Microbiology, where they looked into different transitions that can happen from a healthy microbiome to a so-called unhealthy microbiome states. And depending on the microbiome transitions, you could have different sorts of group variations or within group variations in different stages of disease. And at the same time, what they could all conclude is in many of the diseases, the transition or many of the diseases are actually characterized by the Anna Karenina principle of the microbiome. That is, all healthy microbiomes are alike and all dysbiotic microbiomes are dysbiotic in their own way, or dysbiotic can occur in various manners. Now you can also see this evidence from the Harverson et al. Paper from Nature Microbiology, where they actually looked upon something called as the healthy plane. Now, what is a healthy plane? Healthy plane is essentially a bioinformatical computed plane, and what they would identify is that the healthy samples were generally closer to the plane, and the unhealthy samples were showing large fluctuations. Similar thing was observed in one of our previous papers, which is the KD et al. Paper, where we exactly showed that all the healthy microbiomes were clustered in a single center, and as the different stages of diseases increased, we saw a progressive increase in the beta diversity or in the microbiome variations. In the Harverson paper, also they could observe that samples which are closer to the healthy plane were much more stable, and the ones that are farther off, like you can see the bottom plot over there, which are showing much more larger variations. In line with this, previous studies by us as well as by other groups have exploited the concept. So in the first study, this study by the HMP cohort in US, they came upon something called as the dysbiosis score. Dysbiosis score are basically a measure of how different a microbiome is from the reference healthy microbiome of a given cohort. And they showed that the higher dysbiosis scores were associated with unhealthy microbiota and unstable microbiota. In one of our previous study, we devised the index called as the Kendall Uniqueness Index, and which essentially again was a member of a type of how a given microbiota, how far was a given particular microbiome from its closest microbiome in terms of the hierarchy of the different species. And what we observed is that this index can be actually used to classify bacterial taxa into different groups. Those positively associated with the index could generally had a higher association with disease, and those negatively associating with diseases like this green group here were showing a negative association with many of the diseases. And using this particular information, we also showed that there is a relative degree there is a relative ranking order Okay, there's some ranking patterns in which certain species associate with microbiome health positively or negatively. So this positive, you can see that this line plots on the left, and you can see that you can actually rank the species based on the consistency in which they rank with different kinds of diseases. Although there are variabilities, and this consistency is irrespective of the age group. But here we also had a problem. That is, with multiple reviews, again, many reviews by us as well as by others, they had shown that a normal microbiome varies significantly across different populations also. And there are region-specific variations in baseline normal gut microbiome makes it in fact difficult to identify consistent disease signatures, as well as the same thing happens with age groups. And more importantly, some species behave differently under different scenarios, like Akkermansia, this one example. So till now, what we knew that there are lots of variations in what a baseline normal gut microbiome should look like. Host-associated factors influence this. Nevertheless, there is a ranked order in the way species associate with health. Many of these health-associated species are prevalent across normal individuals. And more importantly, they show an association, albeit uninvestigated association with long-term microbiome stability. So with this objective, we try to explore this even further. So this is a master's project. So we start to explore this. Let's just take this step forward. Let's just take our understanding a bit more further and see what we find out. So what we try to do here in this paper is, in this study is, we try to create a ranking index of gut microbiome species taking into account 4 properties: prevalence, influence, stability, and health association. Prevalence, which we call as the core, that is, the prevalent, they form the core. Influence and stability, if they are influencing the other members and they are associating with the stability, they are called the keystones. And health association, that means if they tend to associate more or less with health, we call them the health association. These we called as the HACs, the health associated core keystones. And we started on our endeavor to investigate and find this out, and to investigate the robustness and variation, and to identify if we can actually identify functional signatures using which we can classify these HACs or predict HACs. So this study, we started off with around 40,000 gut microbiomes collated from around 122 studies across 34 nationalities. These included 12 longitudinal cohorts, 56 mass disease control cohorts covering 17 conditions. The first step was computing the influence of the species, influence of the different species. So we proposed our own method, which is called the RRR approach, using which what we did was we removed the specific species from the microbiome profiles. We recomputed the abundances, and then we recomputed the beta diversity based on these renormalized abundances. And we tried to predict whether the abundance was predictive of the beta diversity of a given microbiome. So using this approach, which I can discuss after later, we started to actually predict for every study. We took only the normal controls, and for every study, we tried to predict the influence score of every species on the gut microbiome. This we did for all these 72 studies that we considered here. And then for every study, we also took the prevalence of the different species. So species covering present in 70% of the samples and having a ranked influence of greater than 70%, we called as the major core keystones. And this was the rank order that we can see. And you can see some of them are known taxa, like Bacteroides uniformis, Faecalibacterium prausnitzii, Doria longicatina. So this rank order includes some of our usual suspects of a health-associated microbiome. However, we did see some major signatures, so changes in the signatures based on the location. Like you can see over here, in the rural and tribal mixed cohorts, you had Faecalibacterium prevotella, Gamigers, Lachia, isoflavonic convertants, and the Bacteroides on the lower side. Then as you move to the urban mixed cohorts, you see that the Faecalibacterium, Gamiger, they came down and the Bacteroides started moving up. Then as we reached to the industrialized and urban cohorts, you had the Bacteroides completely dominating the microbiomes as compared to the other taxa actually going down. Then we saw in ranked order, we calculated the importances, and then we looked into their effect on the long-term stability. So we had 12 longitudinal cohorts from different kinds of patients from different locations. Unfortunately, we didn't have any rural or urban cohorts in this except one cohort from Malaysia. And then, like the Havers and et al., we computed the across time point beta diversity for every sample. And then we associated that time point based stability, time point variation or time point beta diversity to the microbiome species, abundance of the different microbiome species. Now, as you can see over here, as the microbiome becomes more stable, its divergence from its previous sample will increase, right? And that becomes that is an unstable microbiome which has higher variability with respect to its previous sample. Now, the stability, the species which are associated with stability, if they're associated, they should have higher abundance at the previous time points, right? So there should be a negative relationship. And as we observed over here, 26 of the 47 major keystones, there was a significant association with long-term stability. And this observation was replicated irrespective of the distance matrix or irrespective of the subject types that we considered. We consider subjects belong to the various types, and this observation is replicated. So what we observed was 55% of the predicted influenza species had a significant association with the long-term microbiome stability. This helped us to refine our search and refine our rank order as well. The next thing was to compute the fourth property, that was to consider the disease association. Now, as you can see over here, this was only for a discovery cohort. We validated it in a separate validation cohort as well. As you can see over here, you see the green ones, when they are top, they are mostly associated with the green bands over there. And as you can see over here, there is a significant association. So wherever you see the green bands, that is the major keystone, the 47, you see that the green line is always at the top. That means they tend to have a longer or a much stronger association with health. That is, they tend to be decreased in disease. And the same thing that we saw over here when we actually started doing a meta-analysis and started counting the consistency of the associations. These species, these guys were always higher up in the negative side. That is, they were always higher up or had a significant negative effect with respect to disease. And finally, combining the list of all these taxa, we did a lot of other evaluations also, which we have not shown here. Combining a list of all these three analyses, so 4 analyses, we identified a set of 19 taxa, which were detected as a major keystone in at least 20% of the studies, identified as significantly associated with microbiome stability in our stability-specific analysis, and at the same time, significantly associated with health in our disease-specific analysis, also using a random effect model meta-analysis. And these are the list of those 19. In other words, these were our first picture of the hacks of the adult gut microbiome. Now, the next stuff, I will just take just 3 or 4 more minutes. The next step in our investigation was the crucial one. We have identified the hacks. Some of them are pretty known. Some of them are actually our usual good bacteria of health. But the next thing is, can we actually predict the functional signatures of it? Some of it is also known, but we don't know the full profile of a given hack or a given probiotic species. So to do that, what we decided was, let's not characterize the hacks as good hacks or bad hacks. Let's just compute a hack score. Because we have done these 3 association analysis, we can actually compute a score, a combination of a core influence score, a stability score, as a health score, association score, in such a manner that the score awards species with overall high values across this, as well as penalizes species which have higher variable of one score and lower value of the other scores. So we computed something called as a hack score for the species. Now then what we did was, the next step was to actually identify the functional signatures for this. So there are huge databases of metagenome assembled genomes which are already available. There are these metagenome assembled genomes sampled from a given gut microbiome. And these are available as part of Nicholas Segeta's group. They're available as part of the Magnify cohort. And we collated a list of around 40,000 metagenome microbiome assembled genomes, separate analysis, completely agnostic of the hack scores that we have computed here from a different set of data sets. We computed their functional profiles. And then what we did was, once we computed the functional profiles using different functional schemas, the next step was to use a random forest model that could, and to check if the functional profiles could predict the hack score based off what we have found out earlier. And we are on the final stages of wrapping this up, but we are doing some more investigations. This is the last stage that is remaining. It seems we can. So here what we did was, we predicted random forest models, and there is something that I missed out. In these predictions, we devised these iterations such a way that in each iteration we considered 67 species, only genomes of, we randomly selected 67% of the species. We selected only their genomes and trained the model, and then we predicted the genomes by taking all the 33% of the species. So essentially, this kind of models not only helped us to judge how good the prediction is, but also they help us to test whether our model could predict hitherto unknown species and their hack scores based on just the functional profile. And it seems like we can. So this is using CASI, COG, and KKO. We have also done this for keg modules, keg reaction, EC profiles, and PFAM profiles. And the patterns, which I have not shown here, remains more or less invariant. And right now we are in the construction of a panel of functional markers, which is currently underway, using which we can actually predict a given hack score or a hack score of a given species, whether it's known or unknown. So just the last slide. So current large-scale public data analysis provides a first look at the health-associated core keystones of the gut microbiome. It seems that the hacks are predictable based on certain marker functional profiles, indicating that we can identify specific proteins and genes that tune a species to become a hack. It can be used to identify future hacks for next-generation probiotics from newer populations. And it can also help identify a set of marker functions that can be engineered to build new probiotics in future. So with that, I would like to thank the organizers for giving me this opportunity to present my talk. It's a new project, and I would like to thank my students, Abhishek, who is the primary researcher on this project, Om Prakash, who is assisting him, Saurav, who assisted in the functional validation part. He's here right now with me, and my other lab members and my colleagues in the Department of Information, in the Department of Information Technology, Delhi, and our head of the department, GPS Raghava, for all his support. So thank you, everyone.
Okay. So I think that what I'm told is that all question answer would be at the end after all the 4 talks. Okay. Thank you very much. Very nice talk. So we move to the second talk of the session, and that's going to be delivered by Mr. Sunil Nagpal, who is from TCS India. And I would like to invite Nagpal. And Mr. Nagpal is actually also another computational biologist who is working in the area of microbiome research. And also he is with Tata Consultancy Service here. And he did his MSc research from Biochemical Engineering and Biotechnology from IIT Delhi. So you are also a Delhi person. And his research included developing different kinds of algorithm for analyzing the data which are coming out from the NGS and microbiome, as well as doing a biomarker discovery and using the machine learning and network biology, and also NLP. And that's how he takes his research further. Today, he's going to talk to us on a simple keyword play with functional features of the microbiomes of interest, maybe some of the NLP that we learned. Thank you.
Thank you. Thank you, ma'am, for the kind introduction. Really enjoying all the sessions here since yesterday. And I hope I just keep it simple and make it interesting for all of you. So as very kindly introduced by ma'am, that I also have some interest in NLP, but sometimes simple problems are what keep us awake, and we try to get some simple solutions to them. So that's why I have titled my talk as a simple keyword play with functional features of microbes of interest. And while I would like to talk about where I am from, I'm from Tata Consultancy Services, which is presumed to be a IT consultancy company, but it's also doing a lot of microbiome R&D for the last 15 years. But we can certainly talk about what we do and where we do and how we do perhaps after this session. And I would directly jump to the talk and maybe just bring this picture, which might be familiar to all of you, maybe not in this time, but in the past. When was the last time you took a scientific book or a paper in its physical form and tried to probe it for some keywords or your functions of interest or microbes of interest, and see or underlined, and this is my microbe that I was trying to find out in this particular paper, and this is the function I was trying to probe. Perhaps this time is in the past, because at that time we were trying to look for the keywords physically in those physical documents, but the time has certainly evolved into this current stage where computers started assisting us into searching for those keywords. You might remember doing a search on NCBI or on Google using your keywords and trying to find out all those documents that you want to read, the papers, basically, or the books. But also with time, we got more stressed with the kind of outputs we started getting. The documents are now too much in number. Even if we use the keywords, the results are in such huge numbers that this is the state we end up getting into, right? that how can I read all those documents? And this is where a very simplistic technology of natural language processing came into being. And it also helped us to sort of in an automated way, try to infer what is in those books or in those papers. And with a very simple idea of the bag of words in the past. This is a technology of the past, I would say, as compared to what we are witnessing in the LLM space these days. But in the past, the bag of words was sufficient enough to, say, create those metrics or those embeddings and give you with certain probabilistic topics in those papers that you might like to read and relate how various documents are related to each other. And perhaps those are the bunch of documents that you would like to focus on. And just like it is emphasized here that there are trillions of stars in the universe and trillions of microbes in the human body, which is very much a fact. If these are the overlapping topics that you want to probe, topic modeling can certainly help you into zeroing on certain topics or themes of interest. But why am I consistently talking about this when we are here in a conference which is much focused on microbiota or microbiology? Recently in 2020, actually, when COVID was on the peak, a very simple thought came into mind that why are we not perceiving genomes as typical documents, which have certain words like the annotations that genomic assemblies give us, genes, the codons, and various other words that you can perceive in an analogous world, that those genomes have words as well. And a combination of those genomic features could be the topics or the themes that you are trying to probe. And microbial genomes being small documents may be still difficult for us to physically read. The computers can certainly help, right? In today's world with DNA-BERT and LLM, the application of LLM and the contextual NLP, things are very much advanced. And certainly the progress has been made a lot. But back in that time, in 2020, we certainly could ask this question and use a very simple latent Dirichlet allocation, which is a very basic topic modeling process in NLP on those millions of genomes which were being deposited to the repositories for SARS-CoV-2. And it turned out that we were in a very unsupervised way. The topic modeling process was never informed of what clades are or how the evolution of virus is going on. It could very easily infer, not easily, but certainly it could, with a certain degree of accuracy, infer that these are the various signatures or the topics in various genomes which conform well with the clades that were being identified. And also it could infer what are the mutations which are evolving over time. Again, why am I talking about this as well? Right. The topic was a simple keyword play. Why I'm talking about topic modeling and certainly some way of progressive NLP, which doesn't seem that simple to talk about, right? I'm actually here to talk about certain problem statement at the same time that came to my mind. And we were trying to solve this problem that the tumor is a stressful environment, right? The microbes, and there is a lot of talk about cancer and microbe interface. So when those microbes interface with that tumor, there will be a lot of stress they might have to go through. The nutrient stress, because the tumor is highly proliferative, the oxidative stress, because there is a redox imbalance in the tumor, and there is a physical stress because of the osmotic imbalance. And there is an acid stress as well because the tumor surface is highly acidic. So the microbes might, whenever they try to interface with the tumor, there might be a lot of stress they need to go through. And if they have to respond to that stress, they will have to go back to their evolutionary stress response pathways. So how does the response of microbes to that stress create a collateral damage or a collateral benefit for the tumor, basically? How can we, so this was just a hypothesis, a perspective which recently got published. It's all perspective. There's no computational analysis in that. So while we wrote that perspective, the question was, when we know that there are a lot of genomes available, say on public repositories like NCBI, and we know that there are many microbes speculated to be associated with tumor or speculated to have cancer or anti-tumor response, can we not just go and try to do some instant keyword search and find out that for our microbes of interest, which are also our keywords, what are the functions that could have been associated with stress response, and whether there is a significant differential comparison possible between those microbes of your interest. If it is not making sense, it might slowly start making. So despite all those genomes which are there on the public repositories, I could not do a foundational query that what are all those microbes which could express or could have a protein toxin associated expression possible, basically. There are a lot of genomes. Can I instantly find out all the microbes with toxins and how do they compare functionally? Or say, what is the toxin annotation specific profile in Lactococcus and Enterococcus? Can I visualize that? And can I do some interpretation of why Enterococcus is bad as compared to, say, Lactococcus? So that led to just a simple this keyword play with all the annotations which are there for all the microbes on NCBI or other public repositories. And if I just say plug in, I created a sort of a suite for this. So if you just write a keyword, say toxin is my keyword of interest, and I want to search in, say, Lactococcus and Enterococcus, how the toxin-specific gene copy number profiles or PFAM profiles or any annotation of your interest is varying, can I just visualize it quickly, basically? So the problem statement goes back to, again, that I was not able to quickly do the profiling. So this sort of pipeline that I prepared helped me to just do a simple keyword play with those microbes. But then this is not still sufficient because there is a manual interface involved in terms of probing which sort of toxins are actually more prominent in the genomes of my interest. So can I just quickly visualize that how whether those toxin profiles or the gene copy number profiles of those toxin-specific keywords are actually creating certain sort of differentiation between these target genomes of interest? It turned out, yes, it does, but still I can't interpret it. Can I feed it to a ML routine, basically, those copy number profiles with those keywords-based searches, put it into an interpretable ML routine, which is based on XGBoost that Marcus discussed yesterday as well. And you end up basically starting to interpret that what are those antitoxin or toxin-specific, keyword-specific genes or those protein families, how are they differentially contributing to the prediction of, say, Lactococcus versus Enterococcus? And as you might see here, that antitoxin-specific PFAMs are more prominent in Lactococcus as compared to many toxin-specific, which is but assumed more prominent in Enterococcus. But then we know that there are certain audiences talking about vaginal microbiome as well. So why not to just do a all keyword play with, say, Lactobacillus acidophilus and Gardnerella vaginalis, basically, and find out all the functional profile which is there in these two microbes. Too many, too difficult to probe. But it turns out that, say, if I do a instant UMAP with all these, whatever is written is true with that pipeline, that you feed in that information, it gives you that out. So the UMAP tends to indicate that, yes, there is some difference in terms of functional profile, but whatever you just got in heat map is perhaps too difficult again for lazy you to interpret, right? So again, put it to interpretable ML routine. And I don't know how reliable it is, open to your feedback, but it turns out that the machine is trying to tell you that there are just out of all that mesh of keywords associated with various PFAMs in those genomes, there are just these 4 PFAM associated annotations which are trying to correctly interpret that this is Gardnerella vaginalis genomes and the other ones are Lactobacillus acidophilus. The first one is a AAA domain specific PFAM, which has been observed to be, I did a basic, what I shouldn't have been doing, but I did a Google search and tried to look for the paper once again. But because somehow to create some confidence in the tool, you have to go back to the roots as well. So it turned out that the CDC48 protein-specific PFAMs are actually critical for the microbes to survive in stressful environments. by creating a protein homeostasis and degrading all the dysfunctional proteins, basically. And similarly, the bacterial Ig-like domain, the immunoglobulin-like domain, are actually very critical for adhesion to the host cell. Now, if you see the interpretation, that these are very highly abundant, these PFAMs, in Gardnerella vaginalis. So on the right side is the Gardnerella vaginalis domain-specific interpretations, and on the left are the interpretations for the Lactobacillus acidophilus. And it seems to give you a good hint about why we know that Gardnerella vaginalis creates a lot of biofilms in the vaginal microenvironment, because it has those functional capabilities to do so. Again, the point being, with a simple keyword search, often in the abstract or the conclusion of your papers, you might read about this microbe was more dominant in X sample or cohort, and the other microbe was more abundant in the Y cohort. So some interpretations using the publicly available genomes can help you find out the functional relevance of your findings as well, if not available in the research papers. And then going back to that problem statement that I was working on, I know that my keywords are stress response, my keywords are competition sensing, because microbes are trying to compete with cancer in that environment. My keywords are also the microbes that I know have been reported in literature to compete with the cancer by or to promote the cancer or to be anti-cancer in their capabilities, basically. So I took those keywords. I created a set of keywords using bacterial competition sensing pathways that I obviously again went through the literature and read through. We can all discuss all those keywords that were fed in, but the keywords were all specific to competition sensing pathways. And then a set of microbes, which you see in green, being known to be anti-cancerous in their capabilities, and those which you see in red being those keywords specific to the microbial genomes, which are known to be pro-cancerous in their response. The functions are still pretty hard to interpret in a heat map, but the clustering tends to tell you that the competition sensing capabilities or the genome profiles or the PFAM profiles, the annotations used for querying basically, can sufficiently interpret or differentiate between the good and the bad bacteria that I just labeled them based upon the keywords that I fed in. And because maybe after talk, you might ask me that there is what kind of genomes did you feed for your ML routine? You can always specify which genome you want to feed for doing the learning, again, just by simple clicks. So the point is, I kept Bifidobacterium out of the training, Streptococcus salivarius specific genomes out of the training. What I call as a training is Lurgo training, and leave related genomes out so that your models are not actually getting biased by the, I just will take 2 more slides to finish. Your models are not getting biased. So you have a fair reliability that your model is just using those competition sensing specific keywords or the functional profiles, is interpreting it pretty good. What you see on the right, basically. And going into the interpretation of that model, the model which is predicting the pro-cancerous and anti-cancerous microbes pretty well, and you end up realizing that a lot of pro-cancerous microbes are actually having a lot of expression of transporters, PFAMs basically, and a lot of anti-cancerous are either bacteriocin specific have expressions, or they have even the redox balance specific expressions, basically. There is a whole lot of discussion possible on this, but the point being, this simple keyword search enabled to interpret what is that functional capability differentially abundant in the pro-cancerous and the anti-cancerous microbes. And certainly there is more stringency possible to leave the related genomes out, like leave the entire clade out, which I was discussing with Marcus yesterday. In the previous training, you saw that the Streptococcus thermophilus was kept out in the training, basically. Streptococcus salivarius was kept in the test, but it is possible that the Streptococcus specific PFAMs could have been biasing your model to interpret correctly the other Streptococcus species. So you can always eliminate or keep a particular entire clade out of your, or a particular, even the entire genome out of your training, and you can then interpret whether the model is performing well or better or not. So I would summarize that for me, all genomes are documents. Of course, all documents are not genomes, but there is an untapped virtual gigantic metagenome existing on NCBI. There are a whole lot fully assembled genomes which can be interpreted, which can be queried. And because we can annotate them the way we want using the functional profiles that we want, we can certainly query them using simple NLP routines as well and associate them with the foundational data science routines and have better way of making functional interpretations, which may be of help for the larger scientific query or creative queries. And I would like to acknowledge all the team members who have been consistently promoting the scientific spirit in a company like IT company like Tata Consultancy Services. On the top left is my R&D center where I used to work for the last 8 years, but now shifted to the top bottom technology advances, corporate world advances as well. And we are now working in a different unit. And that is Dr. Sharmila Mande, my PI, and my entire team here. And I would like to thank you all for being a patient listener, and any questions we can take after the session. Thank you.